Here at Naiparai Bareilly, we have, I hope I am audible to you, Dr. Jitesh.
Yes, Rakesh, sure.
Okay. So we have 5 departments here. Department of Pharmaceutics, Pharmacology, Toxicology, Regulatory Toxicology, Medicinal Chemistry, and Biotechnology, which we have recently opened last year. The student strength is around 100 or so for one batch of students, including PhD. Including PhD, it's around 120
Okay.
for one batch. We have 15 faculties here in all the, including all the departments, and recruitments are going on. So probably we will get more of faculties in the coming future.
Yeah, I also heard that you're building your new campus. So
yes, so we are planning for our campus to start this year. The construction would be started this year, and maybe it will get complete in the next 3 or 4 years, like that. As soon as possible, we wish that we get it as soon as possible.
Absolutely.
The audience for today's lecture is we have our internal students, faculties, masters and PhD students. And apart from that, there are students and faculties from outside NIPER also, all across the country, from Jamia Hamdard, from different universities, different NIPERs also, NIPER Hajipur, Mohali, Ahmedabad, Guwahati, like that.
Okay.
Most of them are online. That's why we see less crowd in the seminar room.
Yeah, and I hope that this is one of the last few online sessions that we have, and sooner rather than later we start meeting the normal way.
Right, right. So I think we can start now.
Okay.
Okay, so welcome you all to the second session of today's symposium event. In the second half of the day, we have the first speaker, Dr. Jitesh Iyer from Sun Pharma Advanced Research Center. Welcome to you, sir, at Naiparai Bareilly. I would invite my colleague, Dr. Sapna, to introduce Dr. Jitesh to the audience. Dr. Sapna, please.
Thank you, sir. A very warm welcome in post-lunch session. In continuation series of the speaker, I, Dr. Sapna Kushwaha, would like to introduce our next speaker, Dr. Jitesh Iyer. Here is his brief profile. Dr. Jitesh did his MS Pharm from NIPER Mohali and PhD from National Institute of Immunology, New Delhi. He had done executive program in business management from IIM Kolkata. He is an immunologist by training and has co-led discovery program in autoimmunity, inflammation, and oncology. He is experienced in understanding drug mechanism by designing relevant in vitro and ex vivo studies, and is actively involved in target identification and validation. He is an expert in compliance and audit readiness. He is having a vast experience of 15-year research in various companies such as Ranbaxy Laboratories, Daiichi, Advenus Therapeutics, Syngene, and Syngene International Limited. Currently, Dr. Jitesh is Deputy GM in vitro biology team at Drug Discovery Unit in Sun Pharma Advanced Research Company, Vadodara. His abstract of talk would include overcoming translational challenges, Spark cases study. Very warm welcome to you, sir. Our virtual platform is all yours. With these words, I would like to hand over to you. Thank you.
Thanks a lot for that very kind welcome, Dr. Sapna. And good afternoon, everyone in the audience, students of NIPER, students of other pharma institutes who have joined in, all the faculty members and delegates. It's my pleasure to be part of this seminar, and I thank Rakesh and the Naiparai Bareilly team for doing such a good job in organizing this and giving me the privilege to talk to you guys. So the topic that I'll be talking about today would be not very different from what Dr. Jitendra Satyagiri had spoken about today morning, which is the translational challenges. I'll just try to exemplify that by trying to talk about a couple of real life cases that we had undergone at Spark. Just give me a minute while I figure out how to share my screen.
I think screen is rolling. Wait for a few seconds or minutes.
Mm.
इसका
Hello. Can you hear me?
Yes, you are audible. Yeah.
Okay, great. I'm sorry for this technical glitch. Just give me one more second while I try to figure out how to share the screen.
Okay.
Do let me know when you can see the screen so that I'm aware of that.
Sure, sure.
yes, we can see see your screen.
Okay, great.
You may just put it in slideshow mode. It's not in slideshow.
Actually, I've put it. I'll just wait till the network catches up. Could you confirm if you can see the screen in slideshow mode?
No, sir, still not.
Still not there because it's showing up at my end.
You can share the entire screen. Maybe when you are sharing, you share the entire screen.
That's what I'm doing. I mean,
Okay.
I'm sharing the. Entire screen. Okay, sure.
And now we can go to slideshow mode.
Okay. Is it visible now?
Yes.
Yes.
Okay.
Right.
Great. Great. So we are back on. Sorry for that minor glitch. Okay, so today what I'll do is I'll try to just go through what are some of the challenges that a pharma company has faced when we try to translate our discovery to the clinic. So when we talk of translational changes, so what exactly are these? What exactly are these challenges? So we put in a lot of effort in basic research. We come up with a very valid hypothesis. We do a whole lot of drug screenings, optimize the potency, the efficacy. But somehow, once we move to the clinic, things just don't translate. So Dr. Jitendra also talked about the valley of death in the morning session, and that very much does exist. There is this huge valley of death for which there seems to be a very narrow bridge to cross that. And there are quite a few efforts which just die. They just die a premature death in this valley. So what do we do as researchers and as industry professional to mitigate this risk? Dr. Jitendra in the morning also did talk about the immense cost and time it takes to develop a drug. And it would become economically extremely unviable if most of the effort ends up in the valley of death. So it becomes all the more imperative as scientists to think about what needs to be done early on so that we reduce the risk of clinical failure. A brief search on the internet would throw you multiple links on what exactly is translational research and where does the process start and end. You must have seen multiple variations of this slide, right? It starts with the basic science and then moves on to translation to human. So that's the first level that we are talking about, wherein you prove your hypothesis in a mouse or a rat or any such model. And then you have to translate that to humans in the phase 1 clinical study. That's mainly safety in phase 1. And in case of oncology, in case of anti-cancer drugs, you also do a preliminary POC in phase 1. Subsequently, you move on to the second level of translation, which is the phase 2 and phase 3, in which from a healthy person, from a healthy volunteer, the effect needs to be gauged in a person who is actually having the disease in a patient population. That's the second level of translation. For all practical purposes, we tend to believe that once phase 2 and phase 3 is over, the drug gets launched and that's the end of the story. But typically it doesn't happen that way. Even once the drug is launched, there is the next level of translation that needs to happen, which is translation into practice, into clinical practice, which is also called phase 4, in which post-approval, how the drug is accepted in clinical practice by the medical practitioners, and are they facing challenges over there. And finally, the last level is at the population level, in which not one or two or a handful of hospitals, but this drug becomes commonplace across the community and across the globe, wherein a particular disease, for a particular disease, these drugs become kind of the standard of care. So that is the final translation to community. So there are multiple levels of translation that we are talking about, and it's going to be way beyond the scope of my talk to talk about everything. So what I will restrict myself today is the first part, which is T0 to T1, in how at Spark we faced a couple of challenges in developing our drugs and how we resolve them. Again, to keep things in perspective, if we go through, this is basically what what was published in Nature Reviews Drug Discovery in 2017. This actually draws out the roadmap for discovery and development. But in no way intend you to go through all of what is written on the screen, but suffice you to know that it is an extremely complicated process in which there are multiple nodes, there are multiple checkboxes to take care of, and multiple directions your research can take. It is coming together of so many variables that at times it can become really overwhelming for people or for companies unless they actually have a road map in front of them, and even that is not really going to help. To simplify these, there are mainly a few say set of challenges which companies commonly face when they actually try or undergo the drug discovery processes, and I've just tried to summarize the key challenges over here. It could be the lack of a unique chemotype or any chemotype related issues. This is fairly commonplace. Safety issues. Again, in the morning in one of the talks, we heard that 25% of the drugs fail clinical trials due to safety issues, and it's a big number. So safety is one of the things which I'll be focusing on a lot in my today's talk. There are also the lack of a good biomarker. How do you actually determine the effect of your clinical trial, whether your clinical trial has really succeeded or not? Then there are commercial considerations, there are regulatory challenges. So there are all of these so-called non-science related challenges which may again lead to which may actually become a problem in translating our research. The two case studies that I'll be discussing today are going to be focusing mainly on safety issues with an involvement of a very of the physicochemical characterization and the importance of that, because that is again an underlooked challenge in translation. AstraZeneca had recently come out with a roadmap for what is the kind of framework that should be proposed or should be worked upon or adopted by the industry in general. It's a very interesting paper. I put the reference here, and I would strongly encourage all of you to read this paper whenever you get a chance. It talks of 5 things which are very crucial for drug discovery. The choice of the right target. That's very important, because if you don't know the right target, you may not end up getting the right efficacy. The right tissue. You don't want to hit the target at all over the body. If your disease is restricted to a particular tissue, your target should also ideally be restricted over there. Right safety. I cannot emphasize this less. Most people focus on efficacy while designing a drug, but safety is as important, if not more, than efficacy, because if you go through the US FDA guidelines for drug discovery, they really don't care whether your drug is efficacious or not. They have all their guidelines converge around one point, which is safety. Your drug, whatever you launch in clinical trials, should not end up killing the patients. That's all the FDA cares about. So safety is important. The right patient. Okay, there's some kind
Sorry,
of
sir.
background noise.
Yeah.
Yeah, go ahead, sir.
No issues. The right patient to help us design the clinical trial and to focus the results of the same. And finally, right commercial potential. This is very important from an industry perspective, because end of the day, you do need to make at least enough money to sustain your efforts. I'll skip through the details, and I do recommend the students to go through this publication once. It's a very detailed uh, summary of what actually a drug discovery company should be doing like. And yesterday we had this National Safety Week celebration in our company, and I'm sure many other places too have had this celebration. So one of the slogans which our HR put up all over the corridors is this: no safety, no pain; no safety, no pain. In very simple terms, it warns us that a safe behavior and pain are like inversely proportional to each other. For many other industries, it is true, but it is equally true in our endeavor, wherein even if the drug is a little bit less efficacious, it should still be fine. But if the drug is toxic, it's never going to clear your clinical trial hurdles. Um, how do you build in safety while you're doing your preclinical evaluation, right? When we are doing target selection, when we choose which target to build a drug against, we focus mainly on whether the target is specific to the disease or not. That is a given. But many a times we may tend to overlook that the same drug target might have multiple effects. It may also contribute to other pathways. So the non-specificity of a target, that is one of the many reasons which may lead to a future safety concern. Going down the line, we select a lead molecule. We go through the screening process, come up with various scaffolds, and end up with a lead molecule. While doing the lead molecule selection, there are certain established pharmacophores which have some reasonable safety profile, and there are certain others which have some reactive or toxic groups. So we have to make sure that whatever molecule we choose to prioritize to go down the funneling line, we minimize at least the known reactive groups or the known toxic groups on that. There are certain groups which have a propensity to form reactive metabolites, which have a propensity to inhibit human ERG, which is a cardiac channel, which has a major safety liability in drugs. So our design should be focused on minimizing these kind of groups. Further on, when we go towards the lead optimization, we have to make sure that apart from the PK profiles that everybody does, we also have a look at what is the kind of PD profile that the molecule has. PD will tell us whether the drug is actually hitting the molecule as expected, or are there some other outcomes that we are seeing. We have to do kind of a broader test on what are all the different outcomes that the at the PD level that we observe. And finally, in the candidate selection stage, good physicochemical characteristics and a high therapeutic index are two key parameters to ensure that we don't face any future problem in this. There are a battery of tests like mutagenicity and safety toxicology tests which are done and mandated by the company, by the FDA at this stage, and they are like extremely important. I'll just move on to one of the challenges that we faced at Spark. This was done, this work was in the late 2000s. And there were a couple of papers, and no, rather multiple papers, which talked about a retrometabolic approach to improve the safety profile of molecules. So Spark also started on this concept, and it was built around corticosteroids. So you all must have learned in your pharmacology classes that stress can induce the hypothalamus to secrete CRF and ACTH, adrenocorticotropic hormones. These act on the adrenal gland and secrete the cortisol. So the outer core of the adrenal gland is where the corticosteroids get secreted. The inner being the medulla is where you get the adrenaline and the noradrenaline. The outer part secretes the corticosteroids, right? Corticosteroids are of two types. All this is textbook stuff. Glucocorticoids and mineralocorticoids. And glucocorticoids exemplified by cortisone and corticosterones. They have a major role in protein and carbohydrate metabolism, and they have a major anti-inflammatory and immunosuppressive effect. Now, this anti-inflammatory and immunosuppressive effect of corticosteroids have been used a lot by drug makers for the last 50 to 60 years. This is one of the pathways which has been exploited a lot. The glucocorticoids work through by dimerization. They bind to the GR receptor, which is intracellular. That leads to activation of the transcription factors, and there's a direct transactivation of the anti-inflammatory proteins. This pathway has been exploited a lot, and there are multiple diseases in which corticosteroids have been found to be active. A few being ulcerative colitis, rhinitis, asthma, arthritis, dermatology like psoriasis, even rheumatoid arthritis. So there are multiple indications in which steroids are prescribed. A whole lot of skin conditions. If you go to a dermatologist, one of the first things they'll prescribe is a topical steroid. But what are the drawbacks? There are a lot of side effects also of steroids. Steroids are not the safest of drugs, so to say. Steroids being very broad anti-inflammatories, they suppress the immune system big time. So there is a susceptibility to infections. It reduces bone density. It increases osteoclast activity, due to which there is a propensity to generate osteoporosis. It reduces skin collagen. It increases the propensity for diabetes and diabetes-like syndromes. It increases the chance of Cushing syndrome, and there are many more. So there has always been a quest on how to create a safer version of the steroids. Now, why do people still use steroids even if it is not safe? The answer is efficacy. Steroids act like no other drug does. And we are still, even after so many years, we are always hard pressed to find a alternative in terms of potency for steroid. When you do an animal study and you compare your say a JAK inhibitor or any other anti-inflammatory molecule with a steroid, you'll still find that potency-wise the steroid is still superior. That's why people are still worried about how to utilize steroids, but the challenge is always safety. It started off with the initial endogenous steroids, which were actually low potency and could never become drugs. The first generation steroids, prednisone, methylprednisone, they did increase the potency, but they were still not very safe at all. That led to the generation of the second generation steroids, betamethasone, dexamethasone. These are much more commonly used. They are way more potent and have improved lipophilicity. They have much reduced mineralocorticoid activity. So that kind of increases your safety profile. Later on, the third generation steroids came into play. These were mostly restricted for local use. And you have things like fluticasone and budesonide as examples in this class. One of the key parameters of these, or one of the key profiles of these, was that though they were very potent, they had extremely poor oral bioavailability. And so the systemic side effects were extremely low compared to the other steroids. But still not low enough. There were still some challenges with this. Sun Pharma, at that time Spark was Sun Pharma, it was still a part of our parent company, embarked on a challenge of creating a retrometabolic approach for steroids. So what's the retrometabolic approach? So it's like retro is like going one step back, right? So you go one step back in the metabolic process. So you figure out what your steroid is going to get metabolized to, and now you use that as your starting point. So what a classical steroid does, is that it has, so you want the local anti-inflammatory activity. You can formulate it as a local, say, cream or something. But once it crosses your skin, it will enter circulation. And these classical steroids have a long circulating half-life. They undergo systemic circulation. They reach other parts of the body wherein they are not really required, because you're treating a topical case, and you're say if it's psoriasis or something. So you just need to apply that on the site of injury. It's local. But because of the long half-life of these steroids, they eventually enter the bloodstream and cause systemic side effects. So the trick to resolve this is to come up with an inactive metabolite and design a version of the steroid which has much shorter systemic circulation based on the known knowledge about the metabolites of this. So if you design such a molecule, which is a derivative of the steroid, rather a metabolite of the steroid, such an approach has been found to give a much shorter circulating half-life of the steroid. And what Sun proposed was, what if we see if this approach would work to give something like a soft steroid. So many such molecules were made, and we went through the usual rigmarole of screening and assays and all. But to cut a long story short, what eventually happened was, when we tested in the allergic rhinitis model of in guinea pigs, we used a nasal formulation of our lead molecule, and you can see that as compared to fluticasone, we got a much better efficacy in this model. We also went on to test the same in a lung edema model, and we could see a much better efficacy of this. It was almost comparable to fluticasone and better than cyclosonide in a lung efficacy model. So efficacy is established. What next? Next is safety, which is what we were after. So this particular graph here, it shows the thymic involution. So basically, one of the key issues of steroids is they are potent anti-inflammatories, right? So they suppress the immune system. Thymus is one of the major organs from which T cells, the immune T cells, are stored and liberated. And one of the key symptoms of steroid overdose or steroid side effects of steroid is the involution of the thymus. So when we checked for this, so S597 was the molecule developed by Sun Pharma. Cyclosonide and fluticasone are steroid competitors. So we found that the ED50 is a measure of efficacy of the drug, was much more improved, 0.094 as compared to 0.3 for cyclosonide, was much more improved. At the same time, the ED50 for thymus involution, which is the toxic effect, was reduced by around 2 to 2.5 fold as compared to cyclosonide, and much more reduced as compared to fluticasone, which led to an overall safety index. Safety index is the ratio of the least highest efficacious dose and the least toxic dose, right? So the safety index improved to around 90-fold with this retro-metabolic approach as compared to 4 to 8 with the existing competitors. But the story does not end there, and this is where a unique challenge came up. So drug discovery, it's like a test match, a cricket test match, in which you have to play a very, very, very long game. But sometimes it gets just as exciting as a T20. Things just change all of a sudden, and when you're at the brink of victory, you suddenly realize that something has gone wrong, and you have to go back to the drawing board. And exactly same such thing happened when we were also happy with this result, and we started to do our safety toxicology studies as a precursor to taking it to the clinic. Safety toxicology studies are done in beagle dogs. It's an extremely expensive and regulated study. And this particular image is an image of our laboratory in which this study was done. So these are beagle dogs, and this is the respiratory apparatus. So these dogs are made to inhale this particular drug, and then multiple safety parameters are observed. What we found was that we couldn't even make a proper formulation of this drug. We couldn't even reach this particular stage that I'm talking about. And when we did the evaluation of this, what we hit upon was that this was the polymorph of the drug that we started off with. Within 7 days, it converted to something like this. So this was a physicochemical issue. The polymorph that we had with us was unstable and could not pass our stability studies. And we could, this just precipitated out of solution. We could never formulate this. So we had to go back to the drawing board and come up with a polymorphic evaluation of the drug. And finally, we came across a second polymorph, which was much more stable over here. And with that, we went ahead with these rat studies. So it's not only that you design something rationally and it works. Sometimes you hit upon like really big challenges. But eventually we resolved that part, and Sun 5 and 7 was taken to the clinic. This is the clinical trial data which I am sharing here. So as compared to the placebo, there were 3 doses of Sun 597 which was tried, and almost all of them had a very good efficacy. So that's one story. The second story is on similar lines but slightly different. So another target that Spark was working on was on cancer, was on chronic myeloid leukemia. So chronic myeloid leukemia is a blood cancer, a form of myeloid cells of the blood become cancerous. There are 2 forms of this. There's acute myeloid leukemia, AML, and a chronic myeloid leukemia. The standard of care for chronic myeloid leukemia is tyrosine kinase inhibitors. Novartis developed a drug called imatinib mesylate way back in 2001. The trade name for that is Gleevec. And Gleevec has become one of the standard of care for this disease. Gleevec is a BCR-ABL inhibitor. BCR-ABL is a protein which is one of the drivers of CML. What happens is this particular protein, it's a kinase, it gets switched permanently on. And it's a pro-proliferative molecule. And when its pathway gets switched permanently on, there is aberrant proliferation and migration of these myeloid cells, due to which the leukemia actually happens. So these myeloid cells cannot stop growing because the pro-proliferative pathway is locked in the on position. And Novartis showed that if you could inhibit this molecule called BCR-ABL, there was a very good resolution of the cancer. So imatinib became the first orally active targeted drug for many cancers, and particularly for CML. And very soon it rose to prominence and became the first line therapy for CML. The progression rate, which was the very poor prognosis for CML before that, but progression-free survival rate increased to more than 80%, which was a major achievement. And it was one of the breakthroughs of the year 2000. But very soon bad news set in, and people started developing resistance. So not all patients are the same. Their genetic makeup is different, and eventually what happened, that primary resistance to imatinib started sprouting. And it became a very serious problem, in the sense that almost 33% of the patients could never get the kind of response which initially it was shown. And many patients became totally refractory or totally intolerant to imatinib treatment. In some other patients, there was initial response, but eventually the drug response stopped. One of the reasons for this was multiple mutations in the BCR-ABL molecule. More than 100 mutations have been discovered as of now. There were substitutions, amino acid substitutions in these mutations, and one of these key mutations, there were actually 7 of these, which has been listed here. These 7 mutations have been reported to be present in most of the cases. 2/3 of the cases have these mutations, and specifically one such mutation, which was a T at 315 getting converted to I, that was very common. That mutation is known as the gatekeeper mutation, and more than 20%, 1 in 5 people who had imatinib resistance had this particular threonine isoleucine mutation at position 305. A phase of rational drug discovery then started. Both multiple drug companies, Novartis, BMS, everyone jumped into the fray, and then came on the second generation of BCR-ABL tyrosine kinase inhibitors. The examples of which are nilotinib, dasatinib, bosutinib. You must have heard of these names. Dasatinib has a very huge market share even today. The potency of all of these were much superior to imatinib. They were active against different versions of imatinib resistance, but they were still not active against this particular threonine 315 mutation, which was almost 20% of the cases. Apart from that, there was still some safety issues with each of these, and all of them got a black box warning. Like nilotinib got a black box warning on QT prolongation. There was sudden cardiac deaths, sudden death due to cardiac arrest. There was severe myelosuppression with dasatinib. Dasatinib is still a very popular drug. It is still used a lot. But there was QT prolongation which was noticed and severe myelosuppression. And even bosutinib. It was a substrate for PGP. Now, PGP is an efflux transporter. Cancer cells very smartly upregulate certain molecules known as efflux pumps. So these molecules have the uncanny ability to pick out xenobiotics. Xenobiotics is anything that is foreign, as in this case, the drug, and throw it out of the cell. So the cell before it, the drug before it kills the cancer cell throws it out. So this was a major substrate for PGP, and it also caused liver impairment. So there were issues with all of these, and none of them were active against 315. Around the same time, another drug by Eriad Pharma came. This was called a third generation, and this was active against P315, and that was ponatinib. So ponatinib received a very, it was touted as the next big thing in CML, and so-called a Gleevec killer. And FDA gave a very early approval for this drug, and it became the darling of the media at that particular time. That finally somebody has come with a true successor of Gleevec, and the problem is resolved, and we have now got a molecule which also cures the T315I. But there was still a major issue with this. So within 2 to 3 years of its launch, the drug got pulled out from the market, and the reason for that was not efficacy. We had shown, the inventors had shown that ponatinib is a very efficacious molecule, and it actually met the real unmet need, which was the T315I, which all its predecessors could not meet. In spite of that, FDA, which had given a very early approval for this, had to go back and ask the makers to suspend marketing and sales of ponatinib, and the reason for that was safety. What was shown was that there was a risk of life-threatening blood clots, severe narrowing of blood vessels, hypertension, hemorrhage, and this was mapped to inhibition of VEGF kinase. VEGF kinase is the vascular endothelial growth factor kinase, and it's one of the key molecules which are responsible for blood vessel physiology, maintaining the normal physiology of blood vessel. This presented a new opportunity in the field, and a new discovery was made. Now, even if you have something which is hitting all the boxes, now they did not check whether it hits VEGF kinase or not, right? So that was the new challenge that the company faced. And it was around about this time that Spark also decided to enter the frame. And Spark came up. I'm just skipping through the entire discovery process. I'm just cutting to the chase, and we came up with a molecule called vodobatinib. And when we did this, there was particular attention to the VEGF kinase profile. The challenge here was to maintain everything that imatinib did, everything that ponatinib did in terms of 315I, and also make sure that we do not hit PEGF. So there was a lot of drug discovery efforts that went into with that thought in mind. And eventually, after around 4 years of research, we were successful in bringing on what we now call vodobatinib, which had a potency on all the Abl kinase family, which was similar to ponatinib. It was less hitting the SRC families less. SRC is again a broad kinase family involved in multiple immune cell functions. And it completely spared the VEGF family of proteins. This is the in vitro profile, and here you can see that ponatinib. So whatever is green it's like a kinase which is being hit by ponatinib. And here you can see that vodobatinib hits way fewer kinases. It's got a far better selectivity profile over ponatinib. So what happens if you have a better selectivity profile is that even if your drug ends up all over the body, it's not going to inhibit what it is not supposed to, or what is not driving the disease. It will inhibit only those kinases which are actually contributory to the disease, and not every other kinase which is present in the body. And this particular chart shows the selectivity profile of vodobatinib against all the other competitors, and eventually we could show that we have a way better selectivity profile, and that would in theory translate to a way better safety profile. And coming back to our T20. So once we thought that we are there and we have resolved this problem, we hit a new physicochemical challenge here. Vodobatinib was a rock. It would just not dissolve. It would dissolve in DMSO, which is why we could do the initial screenings. Initial screenings are all in DMSO. But you cannot add a drug to DMSO and give it to a person, right, in the clinic. You have to make a formulation out of it. And formulations are either aqueous or oil. Vodobatinib would not dissolve in anything. It did not dissolve in multiple aqueous solutions. We tried different pH. It would not dissolve. We tried oils. It would not dissolve. Organic solvents with some known safety things. It did not dissolve. It just would not dissolve. And Spark was almost on the verge of shelving this program, because if you have a molecule which is extremely efficacious, very selective, addresses the problem perfectly, but it doesn't dissolve, how are you going to administer it, right? That was when we took the help of a formulation group, and there was a couple of very bright people in our formulation group who came up with the idea of a hot melt extrusion process. So hot melt extrusion basically means that you take a matrix and you mix your drug with the matrix, and you heat it. So what happens is that the whole thing melts. When your molecule melts, the crystal lattice is broken. And when you now mix that with this matrix and allow it to cool down, the matrix molecules come in between the drug molecules and ensure that the crystal is not reformed. Now, why does something not dissolve? It's because of the crystal matrix, right? You've got a very tight crystal. It does not ionize or go into solution with the solvent. But if you could bring in something which would disperse it and not allow the crystal to reform, you'll get improved solubility. So that is what we did with this hot melt extrusion thing. Think of it as a powder solution, so to say. It does exactly what happens in a solution, but it's actually a powder. We also tried a nano suspension. That was the initial proof of concept we did. We first made a nano suspension of this. So that actually proved that the nano suspension actually got into solution. So that gave the confidence that it's not something about the molecule. The molecule is able to go into solution, but there's something about its physicochemical property, which is when the second idea of the hot melt extrusion was applied. And eventually we made a solid solution, so to say, of vodobatinib, the API. And when we did a PK of that, you can see that it almost immediately gets absorbed, and we get very good pharmacokinetic profile of this, as compared to this was the original API, which absolutely did not show the required PK. Eventually, we use this hot melt formulation, hot melt extrusion formulation, and with that, vodobatinib is in clinical trials and for CML. And as of today, it's still in clinical trials, but we have got quite encouraging data as of now. So this is the current status of vodobatinib, and it's currently in clinical trials. We started a couple of trials. It got an orphan drug designation by US FDA in 2019 and by EMA last year. So basically, an orphan drug gives us the privilege and exclusivity of certain fee waivers. It's a big thing, as you will realize once you enter the industry. It's usually given by FDA for drugs which have a lot of challenge which have a lot of promise to solve a disease which is for which there's a major unmet need. So vodobatinib has also been positioned by SPARC as a first-in-class disease-modifying agent for Parkinson's disease. And this is where lateral thinking comes in mind, right? It's the same pathway of BCR-ABL, which gets upregulated in CML, which is a cancer, which is also upregulated in Parkinson's disease, which is a neurodegenerative disease. So we have positioned the same drug also in PD, which is currently undergoing phase 2 trials for PD. So let's fingers crossed how that turns out. We are expecting results next year. Let's see how that goes. So that is what I have with me. I just thought I'll share these two examples. So what are the key learnings here? So I just want to highlight the importance of safety. The entire vodobatinib story is because of safety concerns. There were 4 drugs which were launched before vodobatinib. Companies had a 10-year lead on SPARC. Still, SPARC could manage to come up with a very safe and efficacious drug. We ran into multiple problems, which was resolved by out-of-the-box thinking. And we did manage to get an orphan drug approval for these molecules. So these are some of the things that as young scientists, all of us should keep in mind. First of all, always focus on safety. It's very important. You can make the most efficacious molecule on the planet. It would even get launched, but it's not going to get sustained. Eventually, people who take the drug have to live. If they die, your drug is dead. Focus on safety. Efficacy is secondary. Safety is primary. And never ever be disappointed that there are other people ahead of you in the race. It's always going to happen. You just need to find out where the unmet need is, and most importantly, think out of the box. And that's something which I am sure NIPER encourages all of you to do. Myself, I'm a student of NIPER Mohali. That's more than 20 years ago. I have very fond memories of being a NIPER student. And I really thank the ministry people and all the deans of all the NIPERs in India for creating and sustaining such a institute of excellence. And I just hope that more and more pharmacy graduates from NIPER enter the industry and help us resolve all the unresolved problems. I mean, I don't have to tell, there are unlimited unresolved problems in the industry waiting for you guys to come out here and solve them. Open to questions. Okay, most important slide. This is Spark. This is where I work. This is present in Vadodara. So photo of our institute, and these are the people whom I have to thank for this presentation. Dr. Rao, who is the head of drug discovery at Spark, Dr. Talwar, who is the head of in vitro biology, as well as the entire teams involved for Sun Finance 7 and whatever. Open to questions.
Thank you so much, sir, for your exciting and engaging talk. I would like to open this session for the questions. Okay, just we have received a question from Abdul Rahman. His question is: in terms of safety, what are the parameters should be considered during drug design?
Very good question. So when you say drug design, I presume that you mean the initial phase when, you got a scaffold and you want to convert that into your lead molecule. So there are certain scaffolds. So there are two approaches to this, and this is a topic of hot debate. At what time should you check for the safety of your scaffold? So one school of thought goes that you develop the scaffold into certain lead candidates and then you screen them. The other school of thought goes that you check or eliminate the scaffold for major safety liabilities upfront. And then you develop only those scaffolds that pass that particular test, which don't have major safety liabilities. There are pros and cons of both approaches. There is no one right way to do, but you should explore what works in your current micro context. But you have to test at some point of time either your scaffold or your derivatives of that scaffold for major safety liabilities. The second thing you have to keep in mind is selectivity profile. Selectivity is again a two-edged sword. You need an optimal level of selectivity. You go too selective, your efficacy will reduce. If you go too broad, you are going to have a lot of side effects. It's like Buddhism, right? You have to walk the middle path. So you have to know your disease pathways to understand what kind of selectivity is required and try to focus on that. The selectivity part. So these are the two things which immediately come to mind. Rule out scaffold level liabilities and make sure that your molecule is adequately selective. Apart from that, there are multiple other parameters which people do. There are a battery of safety tests which form a part of our drug discovery workflow, which we have to do as and when the molecule progresses.
Okay. Any more questions, audience? Okay, although Dr. Jitesh has summarized his talk, but I would like to thank to him. Means he actually discussed how Spark overcome the physicochemical challenges, solubility of the bodabetinib by hot melting extrusion formulation. That is very exciting. So means we all now know how drug move from the translational research consortium to the safety to the patient safetyness, and how we can focus on the key challenges into the translational research. And last but not the least, no safety no pain, no safety and no pain. This is very beautiful lines in the Spark theme that was floated in your industry. With these words, I would like to thank you so much, Dr. Jitesh. And in consequence to the next series, I would like to invite Dr. Ravinder Kondal for introducing next speaker. Over to you, Dr. Ravinder.
Thank you, Dr. Sapna. I hope you all are having a wonderful time and enjoying all these talks by the eminent pharma professionals. Drug discovery is a long and lengthy process and has many bottlenecks. Our earlier speakers has very beautifully explained some of these challenges, ranging from in target identification, validation to inadequacy of the animals, as well as in finding a right biomarker. But still there is one area that is lacking actually, that is a regulatory, getting and regulatory approval. And for the same, I am very pleased to introduce our next speaker, Mr. Anurag Bansal. He is a Niperite, or I should say Niper product. He is alumni of Niper SAS Nagar. He did his master's in medicinal chemistry from Niper SAS Nagar in 2001. And he has more than 20 years of experience in the field of regulatory affairs. Currently, he is working as General Manager, API Regulatory Affairs and Business. He has submitted more than 20 US drug master files, around 250 drug master files in EU, and also a number of other actually DMF in several other countries. He played a key role in getting regulatory approval for several molecules while he was working there in Sun Pharma. His achievements include handling the complete portfolio of Sun Pharma's for sterile PNM molecules and initiating regulatory dossier for drug product intermediate in Sun Pharma. He contributed to the successful completion of various regulatory audits from US FDA, EU, Japan, Korea, Canada, Australia at chemical manufacturing. He received several awards for doing excellent and great work in fermentation facility and handling the portfolio of sterile PNM molecules in US FDA audit. He also mentored more than 40 associates in regulatory affairs or regulatory functions. The title of his talk is actually Regulatory Challenges in Drug Development, where he is going to summarize some of these challenges being faced by the companies or the pharma companies for drug development. So with these words, now I invite actually Dr. Anurag. Over to you, sir.
Yeah, thank you. Thank you, Niper team. Thank you, Dr. Rakesh. So actually, it's really pleasure to have an interaction with our Niperites because I also belong to Niper. As Mr. Jitesh, who just concluded his talk, we are classmates, and it was wonderful to hear talk of Mr. Dr. Jitesh also. So, Dr. Rakesh, right now I am not able to share my screen. So if you can help me, so that otherwise you can start the presentation from their side.
Can we start from here, sir? Can we share from our side?
Yeah, you can please.
Is it visible to you, sir?
Yeah, it's okay. So okay, so can I start? Dr. Rakesh, hopefully I am audible.
You are audible. Just 1 minute. There is a background noise. So I request all the audience to please put off your mic. That creates a lots of disturbance. So please do that and take care of it.
Yeah. Share it.
Okay, sir, please go ahead.
Okay.
With your presentation.
Thank you, Dr. Rakesh. Thank you. So to go to the next slide, please. Yeah. So what is first of all, what is regulatory? What is meant by regulatory? So yeah, next slide, please. So if we define regulatory department is an important part of any organizational structure of the pharmaceutical companies as on date, because it serves as an interface between the scientific and marketing expectations in getting approvals from the relevant health agency to bring safe, effective, and quality medicines in the market. So, but if we have to do business in any of the country, so we have to get approval from the respective regulatory relevant health agencies. So regulatory functions mainly for the human drugs acts in two parts. One is for the active pharmaceutical ingredient and another is drug formulation part. So regulatory function working as in the for the API part is termed as RAPI, and while for the formulation it is termed as DFRA. So regulatory affair API RA is mainly dealing with the generic business, and there is proprietary business. First, I will discuss with the generic business. So regulatory approval of registration dossier ANDA MA is a prerequisite before proceeding for any export or domestic use for the drug substance or drug product to any of the country. This is critical for overall export business as well as domestic business of the company. While proprietary business is a new drug application is a prerequisite for launch of any new drug into the market. Regulatory approval of any IND, investigational new drug application or clinical trial application is essential for initiation of any clinical trial with a new chemical entity in humans. So mainly because right now I am discussing only for the regulatory guidelines regarding in the human, but there are other regulatory functions also available, regulatory in veterinary, regulatory in medical devices, and so on. There are a number of regulatory functions right now involved. And what are the scope? What is the scope and interactions of regulatory? So regulatory affairs, one click more, please. Regulatory affairs scope is mainly to deal with various departments. Dr. Kesh, can you click one more time, please? Yeah. So basically they are dealing, they are the center points and dealing with the various departments: clinical, preclinical, packaging, quality, marketing, supply chain, procurement. Then they have to deal with the regulatory authorities to get approvals and as well as for post-approval licenses. So some portion is with the industry and some portion they have to deal with the authorities. Next. So these are the various departments which with which regulatory affair department interact. Analytical research, then chemical research division, you can say organic synthesis, product development research, formulation division, drug safety evaluation, tox group, manufacturing, quality assurance, clinical pharmacology, and pharmacokinetics. These are the internal functions with which regulatory affair team is interacting, while there are other some external factors also. So from the external factors, it is with respect to the regulatory authorities. Dr. Rakesh, please. Yeah, there are regulatory authorities, then API business customers who are getting the documentations, then API vendors and starting material vendors with whom these are the external factors with whom regulatory department is making regular interaction. So because I am mainly dealing with the API portion, that is active pharmaceutical ingredient portion. So these are the various departments with whom we are regularly interacting: chemical research, analytical research, API manufacturing and quality assurance, product development research and drug formulation regulatory, API business, global material sourcing, and regulatory agencies. So what are our main functions with the chemical research? Finalization of the starting material, whatever we are selecting as a scheme, and then from that scheme we have to finalize what is the starting material and what will be the regulatory strategy. And then there is a process discussion, in what way process has to be developed, what data is required for the regulatory filing, what data is required for the query response. Then similarly for analytical research, there are certain functions like approval of the specifications, method validation studies, pharmacopoeia compliance, similarly data required for the filings and queries. Then with the API manufacturing and quality assurance, EB protocol approval, EB means exhibit batches protocol, whatever DMF batches we have to file, EB data for regulatory filing and queries. Then for product development and research, we are dealing that they are our customers and we have to provide the API information to support their ANDA and marketing authorization applications. And whatever changes are there for the for any change, we have to discuss with them for the business continuity. API business, we have to provide them open parts, TDPs, technical data package to support their business. and response to the queries, customer questionnaires, quality agreements. Then global material sourcing, we are dealing further qualification of the starting materials, intermediates, qualification of API for ANDA, MA, and finally with the regulatory authorities, where we are dealing with for original submissions, variations, updates, response to queries, and as well as for getting guidance with respect to any submission and response. Yeah. So these are the current functions of the API regulatory. So with respect to functions, so building and maintaining a credible relationship with the regulatory authorities with effective written and verbal communication, submission of the drug master files, maintenance of the life cycle of DMFs by filing various type of document updates, amendments, variations, then handling response to deficiencies, whatever agencies are raising the deficiencies in our regulatory dossiers, regulatory support to API business by providing them the various type of documents. And then we have to provide regulatory guidance and global regulatory strategy to R&D for development and to production for manufacturing robust product, updation of regulatory awareness within the organization, regulatory compliance at the API manufacturing site and contract manufacturing sites, active participation during any GMP audits, interface between different business functions, technical functions, and regulatory agencies. Yeah. So what is the role of API in the product development? First of all, there is any identification of the molecule and its business case is identified. Then comes the next, once business case is established, then next section is API development, wherein there is a role of API R&D. First of all, whatever scheme is selected, there is IP clearance, that is clearance from the patent cell. Then our organic synthesis team is finalizing, optimizing the scheme, analytical searches, doing the analysis for the same. Regulatory is also involved in the development to finalize the strategy, what starting material is to be finalized, and what will be the control strategy, and finally tech transfer. Once tech transfer is done, then scale up batches are taken, and in the batches, quality assurance, quality control, manufacturing are involved, and finally it is supplied to the customers. Then comes regulatory body, where we have to submit our dossier, facility inspections and inspection observations, if any, that need to be responded. And during the filing, after filing, there may be certain deficiencies from the agencies, so which need to be responded by the regulatory team. And finally, after deficiency response, we get the final approval of the drug. And once approval is granted, and then post approval, there may be ongoing DMF maintenance. Maybe we have to file certain updates, or we have to go for the life cycle management. So if we see from this slide, API role of API regulatory gets started from very first, that is API development. And once API is development is started, till the API is in the organization, regulatory team gets involved in that API. Any activity with respect to API, regulatory team will get involved. So what are the major submission countries wherein API regulatory submissions are filed directly or through agents? These are the countries like US. Against them, agency's name also mentioned: US, Europe, Canada, Australia, New Zealand, Japan, Malaysia, Singapore, China, Korea, and Brazil. These are the main agencies wherein direct filings are made, while some of the submissions are also made through our agents. Most of these are emerging markets, so for this submissions are made. So what is the submission format? Submission format is ICH CTD format. Yeah, so what is ICH CTD format? In most of the countries, regulatory dossiers are filed in the CTD, Common Technical Dossier format. It is an internationally agreed format for the preparation of drug applications intended to be submitted with the regulatory authorities. It was jointly developed by Europe, US, and Japan. This CTD is organized into the 5 modules. Out of these 5 modules, module 1 is the region specific, while module 2 to 5 are intended to be common for all the regions, and it is derived by the ICH. For US and Euro EDQM, there is all CTD sections are presented in the one file. There is no open, no closed part, and quality overall summary is provided along with the one file, and this complete submission is referred as DMF or CEP respectively. For US, it is referred as US DMF, while for any submission for EDQM, it is referred as CEP. In case of Europe, Australia, previous slide, please. In Europe, Australia, Canada, New Zealand, and Japan, the document is divided in 3 parts: open part, closed part, and quality overall summary. And this complete submission is referred as the DMF. Why open and closed part? Because in these cases, applicant also gets involved. Along with the applicant, we have to provide them the open part, and closed part is being filed directly with the agencies. While for the emerging markets, the submission is generally in the form of open part, and there are country-specific requirements which are along with the ICH CTD format. So the document is shared with the customers, and they file it together as one dossier. Yeah. So this is the CTD triangle, which is divided into the 5 parts. First part is module 1, which is regional specific. Then module 2, which is quality overall summary, and this quality overall summary is basically for the non-clinical, clinical, and quality part. Whatever information is presented in module 3, module 4, and module 5, it is being summarized in the form of module 2. Mainly in the generic medicines, module 3 and module 4 is presented. Module 5 is presented if this is a new chemical entity, that is clinical study. Yeah. So as I am mainly dealing with the submissions of the DMF, so these are the main sections which are included in the DMF. It includes the chemistry, manufacturing, and control information for the drug substance. Section 1 is for the general information, which includes nomenclature, structure, general properties, etc. While section 2, manufacture, it includes mainly with the manufacturing site, manufacturing process, starting material information, intermediate information, manufacturing process validation information, development information. Then section 3 is the characterization, which includes how the API has been characterized and what are the impurities included in the drug substance. Then section 4 is the control strategy, what are the specification and test methods, how test methods have been validated, validations of the analytical procedures, then batch analysis, complying to spec, then whatever specification we have described, what is the justification. Similarly, for section 5, this is the reference standard or material, or you can say working standard information, container closure, how you are providing, how you are packaging your drug substance, all information regarding the same. And while section 7 is for the stability, how the stability has been conducted in the simulated container closure system, what is the how much is the retest period, at what condition is it is the stable, all this is described in section 7. So what is a drug master file now? So what are the reasons to file? A drug master file is a submission to a regulatory authority that may be used to provide detailed information about chemistry, manufacturing, processing, and packaging of the drugs intended for use in human or animals. Because drug master file is filed to maintain confidentiality of the proprietary information, because it deals mainly with the manufacturing process of the API, and this is a very confidential information. We cannot share it with the customers. So hence, drug master file is filed to maintain the confidential property. And this permits the applicant to incorporate the information by reference to a marketing authorization application or investigational drug application or ANDA or prior approval supplement. Basically, one letter of access is issued to the drug product holder, so that they can refer this DMF. Agencies can refer this DMF in connection with this DMF. DMF is previous, please. DMF is also useful to allow multiple applicants to incorporate the information in their application by reference at the same time. One DMF can be referred by number of applicants at the same time. Yeah. So who should file a DMF and what is the approval scenario? There is no legal or regulatory requirements to file a DMF. You can share your information, complete information to the ANDA holder, drug product holder, and they can include complete information of the DMF into their file. But because we have to maintain the confidentiality of the API process, that's why DMF filing is essential. DMF contain information on the chemistry, manufacturing, and controls of a component, that is drug substance or drug excipient. All the information can be part of the drug product application. DMF itself is neither approved nor disapproved. DMF is, because itself there is no review of the DMF unless until it is not associated with any drug product application. There are some exceptions, like in case of some countries, it is independently reviewed, and then once it is approved, then marketing authorization applicant can use that certificate that DMF is approved. It is only reviewed in connection with a particular application. DMF is reviewed using same scientific and regulatory criteria based on application under review. So what are the types of DMFs? There are, for US FDA, there are 5 types of DMFs. First type is type 1 DMF is for the manufacturing side, facilities, operating procedures, and personnel. Now it is no longer applicable. Type 2 DMF generally filed by the industries, which is mainly dealing with the drug substance intermediates, and material used in their preparation or product. While there are other 3 types: type 3 packaging material, type 4 excipient, color and flavor, and type 5 is FDA accepted reference information. In most of the industries, whatever dealing with the API information, they are filing type 2 DMF for the either for the drug substance or drug substance intermediate, or for any excipient with whatever the contents of the drug product. So for that, type 2 DMF is filed, while packaging material, if there is any packaging material associated with the drug product, for those companies are filing the type 3 DMF. Next slide, please. For Europe, DMF filing is a complex market. Their marketing authorization applications are filed in Europe using mainly 4 procedures: national procedures, mutual recognition procedure, decentralized procedure, and centralized procedure. This itself is a big chapter to understand what type of guidelines are followed to follow this to get the approval through these various procedures. This can be presented in a different presentations, and API information for all these procedures is submitted to EU authorities either via the ASMF or CEP. What is ASMF? ASMF stands for Active Substance Master File. This can be filed for both pharmaceutical as well as non-pharmaceutical drug substances. While CEP, this stands for Certificate of Suitability for European Pharmacopoeia Monograph. This can be filed only for those substances which are official in the EPHUR monograph. It means European Pharmacopoeia. These are the various abbreviations which any person come across during their interaction with the regulatory bodies. Applicant part restricted. Okay, it's okay. Regulatory guidance. What are the various regulatory guidance which we are following to submit any regulatory dossiers? So regulatory guidance have been established by the ICH. What is ICH? ICH is International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use. So ICH has divided the guidelines mainly into the four categories. One is Q, quality guidance, which is called as quality guidance, which is for harmonization achievement in the quality area. Stability studies, what are the limits to be established for impurities, solvents, catalysts, and good manufacturing practices. Another is safety guidelines to uncover potential risks like carcinogenicity, genotoxicity, and reprotoxicity. Third part is efficacy guidelines concerned with the design, conduct, safety, and reporting of the clinical trials. While last part is multidisciplinary guidelines, referred as M, that does not fit in any of the QS and E that is coming into the multidisciplinary guidelines, like common technical document format. So as RA API, we are basically going through these guidelines. ICH Q1A, which is stability testing, Q1B photo stability testing, Q1E evaluation of the stability, Q2R1 validation of the analytical procedures, then ISHQ3. Next click, please. Yeah. So ISHQ3A, this is basically with respect to the impurities. Q3C with respect to the residual solvents. Q6A specification criteria. Q7 good manufacturing practices, and Q8 pharmaceutical development. Q9 quality risk management. Q10 pharmaceutical quality system. Q11 development and manufacturing of the drug substance. Q12 technical and regulatory considerations for pharmaceutical product lifecycle management. And Q13 is with respect to continuous manufacturing of the drug substance. This is a new concept of continuous manufacturing, which is also called as flow chemistry. Then ISH M4 for common technical document, what I talked about, the CTD document. M7 for the mutagenic impurities in the pharmaceuticals. M8 M4, earlier it was paper-based copy, now it is coming as the electronic submissions. So M8 guidance is a new guidance for electronic submissions of the dossiers. Then there are certain country-specific guidance. Mainly from the FDA, US FDA, Health Canada. So these are the logos for the FDA and Health Canada. There are other guidance, EMA, European agencies, TGA Australia, EDQM, that is European Directorate Quality of Medicines. These are the logos. So these are only some guidance. There are these are some of the country-specific guidelines. There are a number of guidelines, variation guidelines, and everything, for which means for every guideline you can have the another presentation. So there is a vast scope of the regulatory. Yeah. So now come the main portion, that is challenges. What are the challenges being faced by regulatory departments during any drug development? So first is the starting material selection. So starting material is an important part for any molecule from where we are developing a drug. So as per ISHQ11, it should meet this criteria, whatever guidance has been provided in the ISHQ11. GMP as per ISHQ7 begin with the first use of starting material. So once you are starting the drug, so once whatever is selected, you have selected as the starting material. From that point, GMP is must. Secondly, it should be incorporated as significant structure fragment. We can say N-1 is also significant structure fragment, but it is not considered as a significant structure fragment. That does not mean structure proximity to the API. Term applies to the material which contribute to the final molecule of the active substance. So if suppose a molecule is getting formed from 3 or 4 fragments, these 3 or 4 fragments will be considered as the starting material. Justification of a late intermediate as starting material by claiming it as a significant structure fragment is not considered a valid argument, as this can't apply to any intermediate in the manufacturing process. Starting material should be of defined chemical properties, structure which can be appropriately characterized. It should be appropriately characterized. IR should be we can do characterization by IR, NMR, mass, UV. So there are a number of characterization techniques which NIPA team is already following for characterizing any compound. So this should be characterized. Starting material should be commercially available. So it should be the commercially available chemical is such a chemical that is sold as a commodity in a pre-existing non-pharmaceutical market, in addition to its proposed use as pharmaceutical starting material. Chemical produced by custom synthesis are not considered to be commercially available. Suppose there is a material which is being used only for a particular drug, that cannot be considered as starting material. If the same moiety has multiple uses, then only generally it is can be defined as the starting material. Non-isolated intermediates cannot be considered starting materials. Starting material specification should be appropriately defined to ensure ISH control at the API stage. Availability information of from the starting material vendor is also one of the challenge, because some of the vendors are not ready to share their information with any of the stakeholders. Second challenge is with respect to the polymorphism. So what is the polymorphism? Any API, any molecule which is existing more than in one morph is considered to be exhibiting polymorphism. So During the studies, we generally come across that crystalline polymorph, amorphous polymorphs, and then in crystalline, there are a number of form 1, form 2, form A, form B, C, like that. So patent literature concerns, because we have to ensure that whatever patent or literature is describing, if we are following that, we should not be, we should be invalidating that patent, and so that no innovator can sue us for that polymorph. Polymorph should be consistent during manufacturing. There should not be any impact of the micronization, because to achieve certain particle size, we have to do the micronization also, but it should not impact the polymorphism. It should remain consistent during the stability. If there is a mixture of the polymorphs, then ratio should be fixed, and it should be defined, and each batch should meet this criteria of polymorphism. And there it also to be need to be studied that whether there is any conversion during fabrication of the drug product, there is any conversion of the polymorphism. Similarly, for isomerism, what are the number of possible isomers? Generally, we are calculating based on the formula 2 raised to the power n. If there are 2 chiral centers, then it is 4. Then if there are 5 chiral centers, number of isomers, you can imagine how many got changed. So for each chiral center, you have to define the control strategy, how you have said that this is the only possible isomer as the final API. We have to come across with the appropriate control strategy. We can have that single crystal XRD possible or not possible. In some molecule it is possible, some molecule it is not possible. Then you have to get consistency that during the manufacturing as well as during stability. These days, mutagenic impurities is one of the important point. What are the mutagenic impurities? Anything that causes a mutation and change in the DNA of a cell. API should meet criteria defined in ICH M7. Generally, for mutagenic impurities, limits are quite low, generally in the ppm, parts per million. And this is being defined based on the maximum daily dose and duration of the drug. Because the limits are quite low, sensitive methods are required for detection at the lower limit. When sensitive methods are required, instruments should also be available at the R&D as well as manufacturing location for determination of these impurities. Control of genotoxic impurities, sulfonate, alkyl methylates are increasing concern area, because these are impurities. These impurities are combined. There is not a one impurity, there is generally 2 or 3 impurities. Then genotoxic limit applied to the combined impurities, not a single impurity. So these are becoming increasing concern areas. Nowadays, another hot area, nitrosamine impurities. What are the nitrosamine impurities? This refer to any molecule containing the nitroso functional group. These impurities are classified as a cohort of concern, which is a group of highly potent mutagenic carcinogen that have been classified by WHO, International Agency for Research on Cancer, as probably human carcinogens. These limits are quite low. While mutagenic impurities limits are generally in the ppm, nitrosamine impurities are generally in the ppb, parts per billion. So the acceptable daily intake has been defined by the regulatory authorities for these impurities, and then the limits are defined considering maximum daily dose of the drug. And these may be possible due to the process of API starting material, because nitroso group using sodium nitrate, nitric acid, nitrites, anywhere we are using nitrate, then these impurities become possible. Then there may be cross contamination, because these are in the parts per billion. If there is any possibility in one drug and in the same equipment wherein this drug is getting manufactured, even after cleaning, there is a possibility in the second drug it may come. So cross-contamination, uses of recovered solvents, etc. Other similar concerns of sensitive methods and availability of instruments as of mutagenic impurities is also being for nitrosamine impurities. Now, regulatory guidance. Number of regulatory guidance are available as of now. So we have to keep in mind every regulatory guidance, country specific regulatory guidance. There are differences also in the country specific regulatory guidance. If the one regulatory guidance is defining a change, it may be annual reportable change. You can implement the change. Other guidance may say that we have to first submit to the agency, then get approval. So there are differences, assessor to assessor variation. One agency may accept one moiety as the starting material, another agency may not accept the same as the starting material. Regulatory guidance are continuously undergoing amendments and supplementation for bringing stringent controls. Emerging countries also now coming with bringing new regulatory guidance. Recently, South Africa has changed their guidance. Earlier, they were not requiring the closed part of the DMF. Now they are also asking for the closed part of the DMF. Brazil is coming with a new guidance. So emerging countries also bringing new regulatory guidance. So this is also becoming regulatory challenge as of now. Regulatory compliance. What is the regulatory compliance? Whatever we have filed, that need to be followed at the manufacturing locations. Process validation need to be done, need to follow the commitment made in the submission strictly, and any change need to be reported through the appropriate regulatory procedure applicable for the respective jurisdiction. Audit is being done by the API business customers. Audits are being done by the regulatory authorities, FDA, US, MHRA, UK, TPD Canada, PMD Japan, and Visa, TGA, Mexico, etc. Nowadays, data integrity is also one of the point which need to be ensured for the regulatory compliance. Because what is the data integrity? That data integrity, whatever we are doing, that need to be immediately written on the paper, official paper, by the person who has done this. If this is being done by a different person and being written by the different person, then it is also a case of data integrity. So data integrity need to be maintained in the manufacturing locations. This is also one of the regulatory challenge across the industry today. Now, because of cost competitiveness, we have there are a number of variations we have to file. Process is getting changed so that we are cost competitive. Then scale is getting changed. Manufacturing sites are getting changed. Agencies are bringing more monographs, so we have to be pharmacopoeia compliance. Specifications are getting changed. Starting material source has changed. Process change is done for the starting material. So there are a number of changes for which we have to do the variations these days to meet the business requirement. Yeah, this is also RA professional. Global competitive. We have to meet competitive with the global, because with the professionals which have good background related to the development, life cycle management, regulation of the persons who are well aware of these guidelines, their availability is very difficult as of now. They must closely track what are the regulatory changes, what are the new guidelines. They should be effective communicator. They should have good English communication as well as, and they can provide regulatory awareness in the organization. Nowadays, retention of the talent manpower, because every agency, every industry needs talent manpower that is a good manpower having good understanding of the regulatory. So it is a global competition, not within the India, it is outside of India also. Industries are demanding for the talent manpower. So all this is from my side. So there are various challenges in this. Thank you. If there is any question, the session is open for question and answers.
Thank you, Anurag sir, for such a wonderful, insightful and informative talk, and giving us an overview of the regulatory affairs, the various challenges that are being faced during the regulatory challenges that are being faced during this drug development. Now the session is open for question and answers. Anyone who has any questions, actually, they can either directly ask, or you can also type in the chat box and I can ask on your behalf. Any question from the audiences? Yeah, please.
Thank you, Anurag sir. It was like wonderful talk. My question to you is that you had talked about the genotoxic impurities in your last slide. So
Right.
what are the sources of those alkaline methylates and sulfonates?
Because if for methylates, generally, if you are using methane sulfonyl chloride in the process, then from methane sulfonyl chloride, if you are using methanol, it.
Okay, so means
Water.
they are made from the solvents, right?
They are mainly interacting methane sulfonyl chloride interaction with the lower alcohols.
Okay. Okay.
Similarly, for tosylates also, paratoluene sulfonic acid interaction with the lower alcohols.
Okay. And my second question is, what is the scenario of like regulatory challenges in Indian pharmaceutical market?
Yeah, India pharmaceutical also nowadays India is also coming toward the ICH, and they are also bringing guidance to meet the ICH requirement. Although to meet ICH requirement, that there will be a concern area with respect to the increasing cost, because once you start developing for the ICH, definitely you have to bring the purifications in the molecules, and which increase the cost of the molecule. So although Indian industry, Indian drug regulatory authorities are also thinking about the ICH criteria still, but there is a vast area which need to be worked for meeting this ICH criteria.
Okay, so we are like more closely compliance with ICS, not with FDA, right?
No, FDA is follow. What I understand from your question, you are asking for the US FDA, right?
Yes, yes,
Yes,
yes.
okay. So you have the US FDA is also asking for the ICH criteria. If you are following
Okay.
ICH criteria, then US FDA criteria is automatically getting fulfilled. I agree. US FDA for submission of a DMF, there is a GDUFA Act, according to which we have to do various submissions. There are certain requirements that have been provided, but that are also in line according to the ICH requirement.
Okay, thank you so much, sir. Now I got cleared my doubt. Thank you.
Yeah. Thanks a lot. Thanks to my NIPER fellow, and best of luck for your future.
Thank you, sir. Thank you. Now I would like to invite my colleague Dr. Gopal actually to introduce our next speaker. Over to you, Dr. Gopal.
Thank you, Dr. Ravinder. Now I am going to introduce our next eminent speaker from the industry, Dr. Nimesh Bhaskaran. Dr. Nimesh Bhaskaran is currently working as an assistant director in Jubilant Biosys, Bangalore. Dr. Nimesh Bhaskaran obtained his PhD in 2003 from Karolinska Institute and Ludwig Institute of Cancer Research, Stockholm, Sweden. He also did 5 years postdoctoral research at the same institute. He started his industrial journey in 2013 in Daiichi Sankyo India Private Limited, Delhi, India, as a group leader in the division of pharmacology and toxicology. Further, he worked as a principal investigator and is presently working in the Discovery Biology Department at Syngene International Limited, Bangalore. He is having more than 9 years of progressive experience in research and development in the pharmaceutical industry. He is instrumental in identifying, evaluating, and developing pharmaceutical and biotechnology prospects for new business opportunities. Also involves in establishing commercial strategy, implementation of lab automation, data integrity, and business continuity plan as a part of strategic initiatives. A most important achievement in his credit include as the lead role in an initiative to standardize the human protein microarray technique in collaboration with the Life Technology USA that was designed for biomarker discovery. Dr. Nimesh had been acclaimed with the Team Excellence Award, Syngene International Limited Award in 2019. His title of the talk is E3 ligase biology and their utilization in protein-based drug discovery. With this short introduction, now I would like to invite Dr. Nimesh Bhaskaran to deliver his talk. Over to you, sir.
Thank you. Thanks a lot, sir. So am I audible and visible?
Yeah, yes, sir. Yes, sir.
Yeah, thank you. First of all, such an elaborate, it was not a short one, it was a quite elaborate introduction. So thank you for that. And the second thing is, so there was a lot of things in the introduction, and I'm not going to speak about any of those. I'll be speaking about something different, which we have been working on, which I had worked on, and I wish to pursue further as well. So there is a request, because there might be few animations. So I would request, I had already requested Dr. Rakesh to project the slide at your end. So if that is possible.
Yes, sir. We received your slide and now we'll be sharing from our end.
Okay, that'll be really great. Thank you, sir.
Yeah.
Thanks a lot. Yeah. So I'll start with this. So as Dr. suggested, the topic of my presentation is E3 ligases, biology and utilization in PROTAC-based drug discovery. So
okay.
This is
can you see, sir?
Sorry.
Slides
so this is the first slide, sir. Introduction slide. I mean, not the introduction, the first slide. Yeah, I'll go by slide number. I'll definitely let you know once, because this doesn't have a slide number. I'll tell you the slide number next time on. I mean, as I move on.
Sure, sir.
Okay, thank you, sir. Yeah, so so I'll be talking about this E3 ligases and how we can utilize this E3 ligases in drug discovery. So as I was telling, this is a pretty new platform, and this is recently people have been using it. So from let's say from 5 years, people have started exploiting this. So this became interested recently in 2019 onwards, because 2 of the molecules have already crossed clinical phase 1, and then that's when people realized like we could use this kind of a technology and a platform to make drug discovery. So I'll just give you a touch base. So I will not go deep into this because, to be frank, I also don't know much about it. This is a pretty new science which we would like to further exploit it. So on slide number 1, yeah, please, sir. Okay, so I'm on slide number 1, which shows the current overview, where I'll just give you a brief introduction. Am I audible? Sorry.
Yeah, yes, sir.
Okay, sorry. Yeah, well, I will just give you a brief introduction, and I will just let you know what E3 ligases are and why do we need to go into this space of E3 ligase, and how people have exploited E3 ligases as a drug discovery platform. Because till now, not many people have used this E3 ligase space to make as drug discovery. And then I'll go into the PROTACs. What are PROTACs? And how PROTACs have been used or utilized along with E3 ligases in the drug discovery. And then further, I would like to just give you an overview about when you consider or when you make a molecule as a PROTAC, what are the different things which has to be considered, like what is a warhead and how do you select a warhead, what E3 ligases, because there are lots of E3 ligases, what E3 ligases, not every E3 ligase work together as a PROTAC, so what E3 ligase you should look into. And I'll just give you some examples of the published data here. So it's already in the public domain, so I'll just take examples from there and how people have worked on this. Another concept which I would like to go again along with PROTAC is a ternary complex formation. So this is something which has come up again, different from the conventional drug discovery where people look into the IC50s or how well the drug is binding to the target protein with the KDs and all those things. That more than that, what people have observed or people have published here is the formation of a ternary complex, which means like you have your target molecule and you have your E3 ligase. Basically, the PROTAC has to bring together these two molecules, the target protein of interest, as well as the E3 ligase in forming a ternary complex. So that is more important than just the binding. So I'll give you some examples on the published data, how people have worked. Linker length. So once I start explaining about the PROTACs, you will understand what the linker length is and how the orientation of the linker is important for that. And said all these things, I would also give a brief idea about what are the advantages, because there are lots of advantages of the PROTACs when you compare it with the conventional drug discovery. And also, since it's a new field, not many people know about it, there are lots of clinical challenges as well. So I'll just take you through as a brief through all these points, and then let you with a proper conclusion, like how we can use, because as I said, this is a new area and a lot of things has to be still performed on this. I'm on slide number 2. So as an introduction, so conventionally, if you take from last 20 to 30 years, so a lot of people have been working on the drug discovery as a conventional strategy, where you're developing a molecule which has a high affinity ligand, which binds to or inhibits a kinase or any enzyme, for instance, and then that molecule is further developed as a drug-like properties, and then we release it as a drug, or we go with all the other DMPK properties and toxicology properties, and then we release it as a drug. So this has proved successful. I mean, especially in the cases of cancer and all those things, where there are lots of kinase inhibitors and the enzyme inhibitors, which are already in the market and they're working pretty well. But with the human genome sequencing and a lot of research going into, we have come across a lot of proteins which are undruggable. What I mean by undruggable is it doesn't have a catalytical site, or it doesn't have any of those properties. It might be a transcription factor, or it might be just plain molecules. But there are published data with a lot of genetic or genome based or the protein based, which people have shown really like these molecules also play a role in the disease development. So how to target these molecules, which doesn't have a catalytical site, how to target these molecules? So that's always been a question in the modern day drug discovery. So that's one of the questions. And the second question is, as we are moving into more and more research and more clinical data coming out, people have seen like most of these molecules, when it goes to the phase 2 or phase 3 and beyond phase 3, these molecules become suboptimal, that their efficacy goes down. And more importantly, there are lots of resistance, especially against the kinase inhibitors. There are lots of resistance which has been developed. A very good example is EGFR. So EGFR inhibitors, slowly with the mutations and everything, people have seen the efficacy of the EGFR inhibitor is going down in the clinics. And then you have different proteins with different mutations. BTK is a best example, the kinase protein, which has led to mutation. And this muted protein or muted enzyme is not being enabled by the small molecules. So one of the best approaches, what we need to do is we should have an alternative approach so that we can remove this protein from the cell, not just by inhibiting them. It will be very good to remove this protein from the cell. So at genome level, we have pretty good tools like siRNA, CRISPR, all these approaches works pretty well. But when it comes to drug-like molecules or drug-like properties for this, it is very difficult. People have worked with siRNAs and CRISPR, but to deliver it into the cell or deliver it into the body and to have target specificity and all those things, there are huge problems which people are facing because of their structures, secondary structures, the large molecules, lipophilicity, lot of issues with those. So it will be good to have a small molecule which can work like an siRNA or a CRISPR, where you can remove the protein from the body. So that's where this all thing about PROTACs and everything came into the picture. And moving on to slide number 3. So ubiquitin proteasome system. So this is where everything started. So ubiquitin proteasome system is something which we have inside our body. So that's where every protein in our body has a half-life. So after that, the protein has to be degraded, and so that the new proteins can be formed. Otherwise, you will have the same protein and it is not going to be efficient. So ubiquitin proteasome system in our body takes care of that. So any misfolded proteins or any proteins which has gone beyond its half-life is degraded through this pathway, and then new proteins are being synthesized, translated, or transcription translation happens. So what is ubiquitin proteasome system? So ubiquitin proteasome system contains 3 major enzymes. I don't want to go much deeper into the details, but I'll just take you through simple. So it has 3 major enzymes in the ubiquitin proteasome system. One is the ubiquitin activating enzyme. That is what is shown in the E3. Just a sec. Yeah. So ubiquitin proteasome system, which is shown as ubiquitin activation enzyme, which is shown as the E1. Ubiquitin conjugating protein, that is known as the E2, which is shown below. And the third one is the ubiquitin ligation, which is the E3 ligases. So the whole phenomena or the whole process works very simple. First, you need to activate the ubiquitin, which is an ATP dependent process. So that is taken care by the E1. So once the ubiquitin is activated based on the ATP, that is transferred onto the E2, which is the E2 binds to the ubiquitin. And then once the ubiquitin is bound to E2 and activated, E3 recognizes this and transfers the ubiquitin onto the substrate molecule. So whichever could be the substrate. And that specificity is brought about by the E3 ligase. So the E3 ligase is the one which decides which protein has to be degraded. So once the ubiquitin is transferred, it leads to multi-ubiquitination on the substrate. This is a continuous process. And once it is multi-ubiquitinated, this substrate protein is recognized by the 26X proteasome system, which is inside our body. And that leads to that goes and binds to this protein and leads to the degradation of this protein. Well, there are various other mechanisms as well, depending upon the ubiquitination chain, whether they are degraded or not. But to keep it simple, once it is multi-ubiquitinated, these proteins are degraded, and then the degraded molecules are released as small fragments, and the ubiquitin is recycled, which means like the same ubiquitin which was bound to the substrate protein earlier can be recycled so that it can go as a cycle. So one ubiquitin can lead to the degradation of multiple substrates of the same type, depending upon the E3 ligases. So that's how the whole system works in. And so this is a ubiquitin cascade, which I just got it from the web. So what it wanted to show is the specificity of the E3 ligases. So if you take a body, so I'm on slide number 4, sorry. So when you take a body, any human system or anything, There are generally only one E1 ubiquitin ligation, E1 activation enzymes. So that is only one E3 E1 ubiquitin activating enzymes, couple of 2 to 3 or maximum 6 E2s. But when it comes to the E3, as I was saying, the E3 ligase is the one which gives the substrate specificity. We have 600 of E3 ligases. And if you look at the pyramid, you look at towards the right side, the specificity increases as you go down. So the E3 ligases are the one which gives the specificity of the substrate, which substrate to bind and which one to take it forward. So each E3 ligase, like there are 600 E3 ligases, and each E3 ligase can degrade more than, let's say, 50 to 60 proteins. And that's how the whole system works. We have only one E2, one E1, might be 6 E2s, and more than 600 E3 ligases. And that's how the substrate specificity works. So people have started using this as a, why don't we use E3 ligases as a tool for the drug discovery development? So before I go into the complete details of the PROTACs and everything, I would like to say how the journey began. So it was how people started utilizing the E3 ligases into the drug discovery. And to be frank, it was all a chance factor. People just observed few things, and from there, the ideas came in and things. So it was not planned or something like that. Most of these were chance factor. So it was early 1992 when they found a molecule called fulvestrant. It was found to bind to and lead to the degradation of estrogen receptors. People didn't know much about this. So this was just kept as a molecule which is new to the degradation of estrogen receptors. So not much research was done after that until 2001. So it is almost after 8 years, people found first concept of PROTACs which can be used. So here, this was the first PROTACs concept of the PROTAC where they use the beta TRCP E3 ligase to develop a molecule. So this was a peptide-based E3 ligase. So I've just put it towards the bottom panel here. So the B panel, which is the E3 ligase which is being used, where you have ovalcillin, that is a molecule which is known to bind to the protein of interest, and you have the IKK phosphopeptide. So they use a peptide linked with a linker as a PROTAC molecule to develop for the E3 ligase. So here you can see it binds, the blue part binds to the beta TRCP, which is the E3 ligase, and the red part binds to the protein of interest. So what it does is the PROTAC molecule, it brings the protein of interest and the beta TRCP in close proximity, which leads to the degradation of the protein of interest. So this is what we've seen. So there are lots of problems with this because we were using a peptide over here. And peptide, as you know, is a large molecule and it has a lot of various other challenges in pharmacological properties or challenges with pharmacological which cannot be developed into the drug. So that's why this didn't move forward. And at 2004, 2004, people just found one molecule, a small molecule, which they thought it's called the Neutralin 3A, which they found was found to bind to MM2, which is an E3 ligase for P53. MM2 binds, works as an E3 ligase for P53 degradation. So they just found a small molecule which could bind to MM2 and inhibit the P53 degradation. So this was the first small molecule concept which came into the research field, where they found like, yes, there is a small molecule which binds to an E3 ligase. Similarly, they also found another polypeptide, which is a VHL based. VHL is also a different E3 ligase, which they found like could be used as a PROTAC in the degradation of androgen receptors. So they just in 2004, they found a molecule. In the next one, in 2008, they again used a non-peptide E3 ligase. So they started developing this Neutralin based E3 ligase in the development of androgen receptors. It is still going on, so people didn't know much about it. And there were other E3 ligases which were developed. That's called the snipers. So snipers is basically specific and non-genetic IAP. IAP is another E3 ligase which they developed, dependent protein erasers. So this is the time which people started developing slowly the E3 ligases, and people started becoming interested in this field of research. Next, in 2010, this is a very important discovery where people found an immunomodulator drug. It is called as thalidomide series. It belongs to the thalidomide series, which binds to an CRBN, an E3 ligase called CRBN, which is known to, and this has revolutionized this area of research because a lot of people even now use this series of molecules to make PROTAC-based drug discoveries. Again, in 2011, there were new technologies which came up called the halotag, where they have halotag fused with the proteins and lead to the degradation. But it didn't work much because it is a quite big molecule, so people still are working on that. In 2012, again, different halotags were used to show the degradation of GST and dihydrofluorate receptors, which again people started working on the molecules. So again, some of the protacs which were developed on the phosphoprotacs, because as you know, most of the kinases, they get activated on once they are phosphorylated. So people started looking into those. Why don't we only degrade the phosphorylated protein rather than the native proteins? Because this is known to bind to both phosphorylated and native proteins. So they started utilizing this, whether we can use this technique to only degrade the phosphoprotacs. So that's when the concept of the phosphoprotacs or disease-oriented kinases can be used, or protacs can be used to degrade the disease-oriented kinases. That's when the concept of phosphoprotacs came in. And it was the time of 2014 and 2015 where the real protacs came into the picture, or real protacs people started working in the real protacs. So this is lenalidomide, which is again a thalidomide series molecules, which they, together with CRBN, which is an E3 ligase, they induce the degradation of 2 transcription factors. And that's when people thought, or people realized, we can involve or induce degradation of undruggable molecules, because these being transcription factors, no earlier molecules were known. And then they involve the degradation of 2 transcription factors, IKZF1 and IKZF3. Further work came in 2015, where they clearly could show that BRD4, which is a major inhibitor or major molecules involved in the cancer, they clearly could show like one of the inhibitors which is well known in the market, that is the JQ01. They use that along with the CRBN to show like this could be degraded and the protein could be degraded here. And since then, 2019, it's, as I said earlier, this is when people really started becoming interested in this part of research, is basically because ARV, so there's a company known as Arvinas. So ARV 1110 was cleared the phase 1. So there was no toxicity, nothing, and it cleared the phase 1 in 2019, and which was known to degrade the androgen receptors. Now they have another molecule which is already known to degrade the estrogen receptors. So these 2 molecules are already cleared. I think there's already in phase 2. And so that's when a lot of big companies became interested. And so there are huge multi-billion money which has been involved in this, and a lot of big pharmas like AstraZeneca, Boehringer Ingelheim, Pfizer, they are doing a lot of research in this field. So this is just to show as a publication, so how publication time, or with the time, how the publication and interest in the PROTAC research has been. So as I said, till 2015, it was slowly going up. But by 2017 and 2019, there was an exponential increase in the research, or the people involved in this research, including the big pharmas, where people got really interested. So, and this has been really a top molecule, or top area of research these days, because people have been really looking into how we can degrade the protein of interest for the disease molecule rather than just inhibiting them. So I'll just walk you through in my next slides. I just walk you through how people have utilized this and what are the really PROTACs and how people have used this E3 ligase space for developing as a drug-like molecule. So just to start with PROTACs. So PROTACs is nothing but it's called the proteolysis targeting chimera. It's together being as PROTAC. So if I'm on slide number 7, sorry to let you know, I'm on slide number 7. So if so there is a blue color box which shows a PROTAC. So there is a cartoon there, and there is a small blue color box where I've just put a PROTAC molecule over here. So how does it look like? So PROTAC has three sites. One is a protein ligand, which binds to the protein of your interest, that is a target protein. And another one, which is the orange one, which is an E3 ligase binding domain. So that binds to the E3 ligase of your interest. And then you have a linker region, which is connecting these two. So this is the basic structure of a PROTAC. In a simplified form, because there are lots of signs and a lot of things. People are still discovering new things every day. So what basically the PROTAC does is it goes, and once it is there in proximity of your target protein and E3 ligase, with the protein ligand domain, it binds to the target protein, and with the E3 ligase domain, it binds to the E3 ligase. So once it is bound to both the target protein and the E3 ligase, it just have to bring these two molecules, that is the target protein and the E3 ligase in the close proximity. That is what we call as a ternary complex formation, which is written below here. It is formed as a ternary complex formation. So once this is formed, and it's quite important to form this ternary complex formation. So once this is formed, the E3 ligase recognizes the target protein, and it leads to the transfer of the ubiquitin, which is there inside our body. So we are not using anything extra. We are using the same mechanism what is there in your body. So E3 ligase is also there in your body, target protein, and the ubiquitin is also there in your body. So we utilize the mechanism there and transfer the ubiquitin over the target protein. And once this target protein is multi-ubiquitinated, it leads to a degradation of the protein. And the best thing is, like ubiquitin, the same PROTAC molecule can be recycled, and it leads to the degradation of other target proteins. I mean, other molecules of the same target protein. Let me be specific. It is not different target proteins, but it is the same target protein, but other molecules it can do. So one molecule can lead to the degradation of multiple same target molecules, and that is very important because it works in if you put it in the pharmacological way, it works in substoichiometric level. So a lower nanomolar potency is enough for a PROTAC to really work. You don't have to really go in micromolar, which is seen due to most of the resistance comes because the molecule has to be in your body for a longer time at a higher concentration. But in this case, the molecules have to be in a sub-nanomolar potency or sub-nanomolar concentration, so it can work as it can do its work efficiently. Moving on to slide number 8. So here, I just wanted to explain how the PROTAC looks like in a 3D structure. So there is a cartoon over here, or a 3D structure, where you have a target molecule and you have the E3 ubiquitinase ligase towards the towards the right hand side, sorry. And so it has, and in the middle you have the PROTAC molecule. So it has 3 parts: the protein ligand domain, which is specific to the target protein, that is the protein of your interest. And you have the E3 ligase, that is the ligand, which binds to the E3 ligand. So this specifically recruits the E3 ligase. And then you have the linker region. The linker region is the one which connects the protein ligand as well as the E3 ligase together. And it's very important, the length of the linker region. I'll just touch base upon in my coming slides. How does it work? The length and the orientation of the linker is pretty important as well. So what overall as a PROTAC, what you need is a affinity. You don't need to bind to your target molecules very strong. Like I said, in the conventional molecules inhibitors, where you need to have a very good efficacy or efficiency or potency to bind to the target molecules. Here people have already shown and observed, like PROTAC doesn't need to bind to your target molecule very strong. And even with this light binding, it gives a pretty good selectivity as well for the target molecules. So the main important concept is what I said is the formation of the ternary complex in this. So I'll just, in the coming slides, I'll just walk you through what are the different characteristics and some published data on that of the PROTAC and how it is effective in the degradations. There are a few points which I've captured. The first one is selection of the protein binding domain. As I said, the one part of the PROTAC binds to the protein of interest, that is called the warhead. And the selection is very important, what you use for the warhead. And the E3 ligase, the other E3 ligase, which has to be, so you cannot use a random E3 ligase, and people have seen like not all E3 ligase work for all the proteins. You have to make sure you're using the right. That could be because of the compartmentalization, how the availability of E3 ligase as well. And the second point is the formation of the ternary complex, which I was specifying. It's very important for the efficient transfer of the ubiquitin ligase, ubiquitination onto the target protein, and leads to the degradation. The linker region of the PROTAC. So how big is your linker region or how small? It is very important for the PROTAC functioning. And also the linker composition. So what are the molecules which are there in the linker region? And that is more to do with the chemistry and the orientation of the PROTACs. So that is the people have observed or also published data. The orientation of the PROTACs or the linker in the PROTACs is very important for its efficient transfer and working as a drug-like molecule. So starting with the first point, that is the importance of the warhead and the E3 ligase selection in the design of the PROTAC. So this was shown very well with Lei et al. So this is a publication which shows the importance of the warhead. So they did a very simple experiment here. So what they did is they used 2 proteins, that is BCR-ABL and the CBL, which is very well known to cause or mutated or translocated basically in cancer. So they are two of the kinases which are involved in the cancer. So they utilize these two proteins, the BCR-ABL and the CBL, and they made two BCR. So they already made a PROTACs or the degraders using two BCR-ABL tyrosine kinase inhibitors, which is known in the market. That is nothing but bosutinib and dasatinib. Sorry, again, I'm on slide number 10. Sorry for this. So what they did is they made two inhibitors or two degraders using bosutinib and dasatinib using two different E3 ligases. One is an VHL based E3 ligase, and the other one is CRBN based E3 ligase, where they used pomalidomide. Pomalidomide is a molecule which is known to bind CRBN based E3 ligase, so that's why they used pomalidomide. So the ones which is made by the bosutinib, so if you see in the Western blot, so they have numbered it as BOS CRBN, and the one which was used to make dasatinib, they numbered it as DAS CRBN. So the one with the CRBN is known as BOS CRBN, and dasatinib is known as DAS CRBN. And similarly, the one which was made with VHL E3 ligases, they named it as BOS VHL and DAS VHL. And what they did is they treated the cells with those, and they just wanted to see how do you see or how well these molecules degrade these two proteins, that is the BCR-ABL and the CBL protein. So they have shown it as the Western. There are different studies. To just simplify it, I'll just put the Western blot over here. So what they noticed is whenever you have an E3 ligase or a PROTAC made out of bosutinib and dasatinib. So these two molecules, when you link with CRBN, that is an E3 ligase, CRBN E3 ligase, and you make a PROTAC out of that, you see a very nice degradation, or you see a very nice dose-dependent degradation of both the proteins, that is the BCR-ABL and the CBL. In both the cases, as you can see towards the left-hand side, there is a clear-cut degradation with those. But the same molecule, when you have it with VHL, the bosutinib VHL as well as the dasatinib VHL, it doesn't degrade to the same extent. From this, they concluded like, yes, the choice of your E3 ligase is very important. Which one would you like to take it further as a PROTAC? So that's what they concluded from this, like which E3 ligase you would like to take as a choice is very important in the development of the PROTACs. Another important, this one which they show is whenever you use bosutinib as a VHL based PROTACs, you're not able to degrade CBL, especially CBL protein is not able to degrade as you can do with the CRBN. So these are the two observations they clearly show, or they clearly saw, like the choice of the E3 ligase which you use to make a developed drug molecule is very important in the development of the PROTAC. And also what you use, either you use it as a bosutinib or dasatinib, because dasatinib here towards the right hand side, you can see at the lower panel, it was able to degrade, whereas the bosutinib along with the VHL, but bosutinib was not able to degrade with the VHL. So these two observations, they clearly made like the choice of the E3 ligase and the warhead is very important when you design a PROTAC. So researchers or most of the pharmaceutical people or in the research, what they do is once you start making the PROTACs, you try to make it with different E3 ligases and different target binding molecules so that you can come up with the best one. So that's how the research has been carried out in most of the pharmaceutical companies. The second one is with the point which I was really focusing on is the formation of ternary complex. And this is published in the paper called Bondison, and where, so they here what they did is they used, so I'm on slide number 11, sorry. So where they used a promiscuous inhibitor, a promiscuous kinase inhibitor that is known as fortinib. So what they did a binding studies with 133 kinases, and that is towards the right hand side on the top panel. They've shown a binding pattern or the kinome binding pattern and the A panel where you can see the red dots over there. Those are the bindings. So in the kinome panel, this fortinib is known to bind to 133 different kinases with lots of potencies, various potencies. And when they made this fortinib into a PROTAC, and they treated, so they made a VHL based PROTAC of this fortinib and they treated MDA. So this was done in an MDA MB231 cells, and they did a tandem mass spectrometry just to understand how many proteins are all these 133 kinases getting degraded by this. So to their surprise, what they found is only lesser than 10 kinases were getting degraded. Not all, even though it binds to 133 kinases, but not all of them were able to getting degraded. So that's raised to a question like, why only 10 of them, even though it binds very specifically to this, only 10 of them is getting degraded, which they did further analysis. And what they found was a weak binder can become also a potent degrader. It doesn't have to be that molecule has to bind to your protein of interest very strongly. Even if it is a very big binder, that can lead to a PROTAC. And that is clearly shown in the C panel down there. So if you see, they made a scale here, and they clearly showed that towards the right hand side, what you see is the binding affinity, and towards the y axis is the degradation, and towards the right hand side is the binding affinity. So there is a molecule called this molecule, fortinib, is known to bind very weakly to a molecule known as P38. But when you look at the degradation, it is able to degrade it. So that's raised the question like, even though it binds very weakly to P38 alpha, how come it is able to degrade? So what they did, they found the binding affinity, which was found to be 11 micromolar. That's pretty high from the pharmaceutical, perspective, to have a molecule with 11 micromolar, a kinase inhibitor with 11 micromolar potency. But when they developed this into a PROTAC, they could clearly see a DC50. DC50 is nothing but degradation constant, and they were able to see like it is much lower, that is 210 nanomolar, which is much lower than that of the 11 micromolar. And these are the Western blots which they did, and they've done other studies as well, which clearly indicates like it is not the binding affinity, it is the ternary complex formation which is important for the development of the PROTACs. So here they have shown on the top panel here, P38 alpha, where they have shown the degradation through Western blots, and also through pull down studies, they were able to show like this molecule, the PROTAC, which is made through fortinib, is able to bind and pull down the P38 alpha. So if there are any questions, please, I would request you to stop me in between and you can ask me, because I might be going very fast, so you can slow me down as well. Hello, am I audible? Hello?
Yes.
Yes, sir, okay. You are audible.
Sorry, yeah, sorry for that.
You can go ahead. We will ask the question at the end of this.
Sure, no worries. So I'm slide number 12, and this is just to illustrate what is the importance or the formation of the ternary complex and how is it important to the degradation. So these are the two different tools, one of the two different tools which are used basically in the PROTAC-based chemistry and PROTAC-based drug discovery. One is a ternary complex formation, where we generally do an alpha screen a lot, where you are looking at the formation of ternary complex. And the other one is an hybrid-based assay. This is a cell-based assay. It's called the hybrid technology, where people start looking at the degradation of the molecule, of the protein, or the molecule of your interest, or the protein of your interest. So I've just shown two different PROTACs, A and B. So the PROTAC A, as you can see here, it forms a nice different alpha screen towards the top panel. Like you can say, there is a nice hill-shaped alpha screen data which you are getting. So that seems like there is a molecule, this PROTAC is able to form a ternary complex, very nice ternary complex, whereas the PROTAC B is not able to form a ternary complex. You don't see a nice graph. So it is a very scattered graph which you get. And when you compare it with their the same PROTAC with their degradation potency, with the hybrid assay, the below panel, you can see that the protein of your interest, as the concentration goes down, it is getting degraded. Whereas in the other PROTAC, which is the PROTAC B, you see a flat line that it is not able to degrade the protein of interest. So this is another example just to show like the formation of ternary complex is very important in the molecule, in the making of the molecule of the PROTACs. Another point which you have to see here is the peak over here where the ternary complex happens, the peak of the hill, when you compare the concentration with the concentration of the degradation, that's where you see the maximum degradation happening. So the peak corresponds to the maximum degradation. So that's something which is very important, which clearly shows like the ternary completion at peak leads to the maximum degradation or the transfer of the ubiquitin of the protein of interest. So then I would like to move to the next topic, which is the choice of the linker or the design of the linker. So in this study, what they did is they again made a PROTAC for BTK molecules. So this is using a known BTK inhibitor, that is ibrutinib, and they used a CRBN molecule, which is the pomalidomide. So they made a PROTAC out of this. And what they did here is they changed the linker region. So they shorten the linker. So towards the table, towards the right hand side, so there is a molecule known as MT540. I'm on slide number 13 again. Sorry for this. I keep on missing it. What they made, the molecule is MT540. And you can see the linker region over here towards the right is 12 for that. And the DC50 for this molecule is 80 nanomolar. As I said, it is in the lower nanomolar potency we generally get. And at 1 micromolar, you're seeing almost 96 of the protein is completely degraded. So when you shorten the linker region in the next molecule, which is the MT781, where they shorten by one series, by this, the potency goes down. The potency is still there, 68.3 nanomolar. And you further shorten the molecule, the potency is completely lost. Like at 8, MT797, there is no degradation. And even MT783, which is around 5, the linker length is 5, there is no degradation in this molecule. So that is very important. So to keep the linker length optimal so that there is enough space to form the ternary complex. And that's what they have shown over here. Another thing which they noticed is they used the same molecule MT540 and they made 2 different molecules. That is, they changed the linker region, 8-atom linker region, and they changed the position. In the 5th position, they made a thalidomide ring of the pomalidomide. And again, at another one, they changed the linker region at 5 and 8. So what they say is when you change the orientation, like in 8 and 12, the potency of these molecules keeps on changing as compared to this molecule. So it's very important at which orientation the molecule is, you're making the linker region so that it becomes optimal for the PROTAC to be working or leading to the protein degradation. Similar studies were done by another group. So I'm on slide number 14. So what they did here is they generated isoform specific PROTACs, which means like P38 is a MAP kinase protein. They have different isoforms, alpha, delta, gamma. So they utilize 2 different isoforms, that is P38 alpha and P38 delta. And they made PROTACs for this, specific for this isoforms. And they named it as, so they used the warhead, which is ferritinib, which was seen earlier, along with the E3 ligase, which is the BHL E3 ligase was used. So SGF alpha is known to degrade the alpha isoform. So that has a 13-atom linker region and an amide attachment to the VHL. And similarly, SGF delta is a 10-atom linker with a phenyl attachment. So the attachments are different and the lengths are different. And they checked how does it affect the degradation of the proteins or the isoform specific. And what they observed was SGF alpha was specifically degraded only P38 alpha. Towards the right, if you see the Western blot, so you can see SGF alpha was only degrading the P38 alpha, whereas SGF delta was also was only specifically degrading the P38 delta. It was not degrading P38 alpha. And the DC50s were mentioned, like P38 alpha with SGF alpha was around 7 nanomolar and SGF delta was around 46 nanomolar. So the question was, why are these molecules only specifically degrading? Because it's just the same and only the linker as well as the attachment is changed. So they just wanted to check the binding affinity. Does these molecules bind properly? So they utilized this P38, SGF alpha and delta. They did a pull down studies, which is in the below. They did a pull down studies with P38 delta, and they saw that both the PROTACs, SGF alpha and SGF delta, were able to bind equally well to P38 delta. Then the question was, if SGF alpha was able to bind to P38 delta, why is it not leading to its ubiquitination and why it is not leading to its degradation? So they've done a lot of studies in this paper, and they show that SGF, when you do a ubiquitination blot towards the C panel, this is the ubiquitination blot, they see that SGF delta, which is in the middle panel, leads to a better ubiquitination. The darker band over here leads to a better ubiquitination as compared to SGF alpha, and that is due to the formation of the ternary complex. They have done other studies and they clearly showed like that is due to the formation of the ternary complex, which leads to the ubiquitination and the degradation of the target molecule. It is not just by how strong do you bind to your target of interest. Moving on to slide number 15. So these are the E3 blazed PROTACs over here. I don't want to go into details because there's a lot of chemistry and I don't understand much. But what I can say is these are the major 4 PROTACs which are being exploited very vastly in research. One is the MBM2, which I said earlier. This is a P53 dependent E3 ligases. People have used Nutrilin, which is a molecule which is known to bind to MDM2, and people have used this as a PROTAC. Another one is CIAP, which is the E3 ligases known to form, Bestatin is known to bind to CAP, and people have used this as an E3 ligases. Very well studied molecule is CRBN, which is a ring type E3 ligases, and people have found like an immunomodulator drug, that is pomalidomide, is known to bind to CRBN, and people have exploited this as a E3 ligase or a PROTAC molecule, which is being currently studied a lot. There are other molecules like VHL based, which is very well known to hypoxia-induced E3 ligase for hypoxia-induced protein. So people have used this as well as a PROTAC to your degradation or the PROTAC-based drug discovery. So these are the major 4 studied. Still, people are looking for other, or research is being carried out to look for novel ones. But to be frank, these are the major ones. If you look into the publications till date, these are the 4 different E3 ligases which people have exploited in the drug discovery market. Moving on to slide number 16. So said that, I've given you a lot of or a brief description about the PROTACs and how E3 ligases spaced have used. So what are the advantages? Why do you want to move into the PROTAC based rather than the conventional drug discoveries? So one of the things is, as I was suggesting, there are a lot of molecules which are already coming up, or the protein targets which are already coming up, which are named to be undruggable. And with this approach, you can always target the undruggable because you don't need a catalytical site or an active site for your target to bind, or a small molecule to bind. It can be a surface binder as well, which can be developed into a very good PROTAC. Because the affinity, one of the main reasons what you need is you need an affinity of your molecule to bind to the target protein of interest, and no catalytical sites are required. So another thing, what with the drug discovery and a lot of clinical phase data which is coming up, just by inhibition is not alone. There are lots of resistance which is being coming up. Proteins are getting mutated, and especially the kinases, and they are becoming resistant to lots of molecules which are already there, which are used in the conventional drug discoveries, or which are conventional treatment for most of the cancers. So we need an alternative approach to degrade the molecules rather than just inhibit the molecules. And again, a lot of proteins like scaffolding proteins, multiple functional proteins, it is important for them to remove. And since this works in a low nanomolar potency, sustained pharmacological effect is required, which means like even if after the drug is removed from the body, a PROTAC is removed from the body, since the proteins are getting degraded, it takes time for it to come back. So it is not just the inhibition, like the molecule leaves the protein, again the protein gets activated. Since it is degradation, you have to start over again. So there is a sustained pharmacological effect. You see a better functional selectivity when you make a PROTAC rather than just the conventional drugs. So these are the lot of, and most importantly, it works in substoichiometric level. Since the same molecule can degrade multiple molecules, so the same PROTAC can be used. So even a lower concentration is very effective. That's why when you see most of the papers or most of the researches which is being done, and even in the clinical studies with Arvinas, so people have seen it works in a lower nanomolar. So the chances of toxicity is very less. So that is one of the important aspects why people would like to move into PROTACs. I'm on slide number 17. So said that, there are lots of advantages. There are lots of, so with advantages comes the clinical challenges as well. So one of the biggest clinical challenges what people have been observed is, since it's a new molecule, there are differential activity in the cells as well as the preclinical model. In the cells, it works very well, but when you take it into the preclinical models or the mouse models or the rat models, the activity changes. So that is something which people have been seeing. So one of the main reasons which I didn't point out earlier is, since this carries the warhead, the linker, and the E3 ligase binding domain, these molecules doesn't fit into the typical Lipinski's rule. So they are the molecular weight for these molecules are generally about 800 to 1200 Daltons. So that's pretty big molecules. It's not lesser than 500. So that's one thing which people have to always keep in mind, that these are bigger molecules than the conventional small molecules which you work with. So that's one of the reasons why there has been a disconnect. But people have been able to overcome this challenge as well, because otherwise we wouldn't have these molecules in the clinic. So more research or more ideas have to be brought in to understand this, what is a differential activity. Since these molecules are big, oral viability becomes a challenge. Going into a solution or let's say solubility of these molecules is a huge issue. So that's something which people have been working on. So you cannot use a conventional carrier molecules which we use for drug discovery. You cannot use that. You need a different altogether vehicle which you can use for the oral viability. And another important problem is blood-brain barrier. So even in the conventional drug discovery, there are lots of molecules which cannot cross the blood-brain barrier. And this being even bigger molecule, it has even challenges of having blood-brain barrier. Even though Arvinas claims like they have molecules which can cross this, but not clearly understood how to move forward. So that's one of the challenges with this. Selectivity, yes, even though they are very selective, possible off-target effects is not very well studied. People are looking into that. So that's something which has been done and people are really looking. And even though as I said earlier, there are around 600 E3 ligases. So, but if you look into the research, mostly E3 ligases or PROTACs are being made out of only 4 E3 ligases. That's VHL, CRBN, CIAPs, and MDM2. So we would like to, or more researchers have to be put in so that people can look into different E3 ligases, especially the nuclear E3 ligases, where they can look into that and we can target more and more transcription factors. So with all these challenges and advantages, as a concluding slide, I would like to bring in and say like, so I'm on slide number 18. PROTACs definitely more challenge. I mean, have opened new chapters for the development of new drug, and it is a novel. So that's definitely on. It's a novel chemical knockdown tool which at the protein level. At the genetic level, we already had these lot of techniques, but at the protein level, we didn't have. So this is a novel chemical knockdown tool which is people have been working on at the protein level. It definitely gives a lots of opportunities, especially in the industry and academia level, which we can utilize or exploit to overcome the current challenges in the drug discovery. With that, I would like to say thank you and I'm open for questions.
Yeah, any question from the audience? Okay, so I hope this is well understood by the audience and they don't have any question. But yeah, of course, you have discussed a lot about the E3 ligases and thereafter development of those into the PROTAC kind of drug therapy. So I just want to know about the E3 ligases, like say, these could be, as you said, these do not follow the Lipinski's rule, having the higher molecular weight. So what is the thing which actually get channelized these kind of molecule in situ, like say drug delivery system? What could be the possible drug delivery system?
No, so the drug delivery system has been the same. People have been utilized this, but there are challenges how to put it into the molecules into the mouse. So the most important thing is delivery is always there, but what you get as a metabolite is a major challenge which people have looked. So the DMPK concept becomes the most important because you really need to understand what metabolites you are understanding. So you cannot use it as a small molecule in the DMPK research. I mean, treat them as a small molecule, but rather you will have to use them as a large molecules. So you'll have to consider these as a large molecules and then do it. So that's one of the challenges. So most of the things are being done, or the delivery system is being used as the large molecule delivery system rather than the conventional small molecules delivery system over here.
Thank you, Dr. Nimesh. Now Director Sir is already joined.
Thank you. So we have Dr. Manish Diwan with us from BIOLEC, and I invite our director, Dr. Yasin Murthy, to welcome him, and then we will go ahead with the formal session.
Thank you.
Thank you, Dr. Nimesh, for your wonderful talk. And it is my pleasure to say a few words about my friend, Dr. Manish, who is an architecture and then making the youngsters into the potential entrepreneurs to make the India as a self-resilience and self-sufficient India. And he's my good friend as well as well-wisher of Naipur Guwahati. And I think formal introduction will be given by my colleagues there. Dr. Manish, I have another program at IIT Guwahati. That's why I came here and I want to say a few things about to my students, about you and your dynamism and your punch about your entrepreneurship element of introducing the entrepreneurship to become how you can become a what you call patardhari, sutradhari rather than patardhari. So welcome once again to the Naiper Library, and then have a wonderful session. Over to Dr. Rakesh. Manish,
thank you so much, Professor Murthy. Always pleasure to connect with you and the fellow colleagues. Thank you for being here. I appreciate that.
Thank you very much. Thank you.
Thank you, sir, for joining. So thanks, Dr. Nimesh, for your scintillating talk and motivating our students. There had been wonderful sessions today, and we discussed so many things about drug discovery and development. But we all know, like for discovery and development, we need funds, and that we get from public sector. So to aware us about that, we have Dr. Manish Diwan with us from BIRAC, New Delhi. And he would be talking about from translational research to product deployment, what are the opportunities extended by BIRAC in terms of funds. So before we move ahead with his talk, let me introduce him to all of you. Dr. Manish Diwan is currently working as a head strategic partnership and entrepreneurship development at BIRAC, Department of Biotechnology, Government of India. Dr. Diwan has been instrumental in guiding several biotech startups to undertake strategic research and innovation. He is leading large national programs for the Government of India efforts to nurture and empower biotech innovation ecosystem. Sectoral lead for national missions schemes, especially Startup India, Make in India. He has contributed to various platforms such as policy reforms, strategic collaborations, including international partnerships, and also serves in national committee and advisory boards in Government of India. He manages risk funding through grant in aid, equity, setting up incubation centers for capacity building, human resource development through integrating a large partner pool of mentors, experts, peers from academia and industry. He has about 27 years of global R&D experience in pharma and biotech sector, from discovery research to product development portfolio. Science administration and project management for both industry as well as for academia. Before moving into public sector in 2017, he contributed to several development of several clinical drug candidates for global drug development at Daiichi Sankyo India, Daiichi Sankyo Japan, Ranbaxy Research Laboratories, Dabur Research Foundation, and University of Alberta in Canada. Also developed technologies during his doctoral research at ICGEB New Delhi and National Institute of Immunology New Delhi, and during his postdoctoral research at Korean Advanced Institute of Science and Technology, South Korea, and the University of Alberta, that were transferred to industry. Trend in pharmaceutics, molecular pharmacology, and immunology like diverse areas. He has led large professional teams in first-in-class and best-in-class drug discovery programs, translational preclinical research, and early clinical development. So with such huge experience and guidance, he is now with us today to share what are the opportunities for funding the research part and development part that is being done by industry and academia, and how government can help us in that. How can we get funds from BIRAC DBT organization? So over to you, sir. Over to you, Dr. Mies.
Thank you, Dr. Rakesh, and glad to be here with all of you, the faculty, the students, the future leaders actually of the pharma industry. And Nimesh, glad to see you too. I missed the morning session. Several of my colleagues, friends participated. Couldn't join you earlier in the day. Excuse me for that. But I'm glad that this kind of recognition is there. And Dr. Rakesh, along with his colleague, has been able to pull out a program introducing you the entire basket, entire journey of discovery to development. And I'm sure since morning you've been hearing about various practical aspects which often we do not get to hear while we are studying. But I think important elements, how the industry is looking at things, what is the expectation from the industry once you tread the path of drug discovery and then to go to development. Very different ball game. As as Dr. Rakesh said, the process of moving from discovery to development, it's longish and quite a bit of resource intensive, and the sector requires people who can actually facilitate this journey. I think the institutional support where you are getting trained, and I think the entire faculty is uh, committed to share the knowledge and prepare you for the next phase of journey. That's part of the enabling environment. What I will try to bring together is another element of the enabling ecosystem, so that you get to know that how, after you attain that knowledge, how you can translate and deploy that knowledge into something tangible as a product or technology. So Rakesh, can I share some slides?
Sure, sure. I will confirm you.
Let me try that. Okay. So, just a second. Let me know once my screen is visible to you.
It's perfectly visible in slideshow mode.
All right. Thank you so much. Thank you. So happy to talk to you. The topic that has been assigned to me is the translational research to product deployment, and what could be the possible opportunities that BIREC has put together, not only for you but for the entire biotech ecosystem, but of course available to compete and avail. Uh, Biotechnology Industry Research Assistance Council, or BIRAC, supports innovation all the way from ideation to commercialization. And the since you are part of the pharma students, know you already understand the most longish and very high attrition in terms of drug discovery or drug development that is kind of unmatched in the other sectors. You would have far better outcome in, let's for example, medical devices, diagnostics, but for the discovery part, in terms of pharma, the success rate is far low. But you also realize that once you are able to bring together a molecule into the market, the rewards, forward rewards, are much higher compared to any other sector. So this particular sector, by and large, has common nuances and common opportunities. The gestation period from initiation to deployment is extremely large. Unlike IT or fintech or e-commerce, where you initiate and within 1,000 days you are able to actually scale really big. You would probably would have heard about the unicorns that India is so proud of. Many of them have come from services sector, IT, and e-commerce, and these have kind of evolved fairly quickly. So that's a basket of opportunity. But from the longish gestation period, like pharma, biopharma, healthcare, agriculture, industrial biotechnology, clean energy, that's a little more resource intensive, and uh, and it stays for a longer duration to put these things together. BIREC is a dedicated enabling agency which supports the entire basket from innovators, entrepreneurs, startups, SMEs to large industry, and it falls under Ministry of Science and Technology under Department of Biotechnology. Uh, so just like you do your master's thesis or PhD thesis for one particular aspect, if that aspect has a commercial potential, So that is where we come, and you can engage with this kind of enabling requirement. So from that position to all the way to commercialization, there are various state-specific uh, facilitations, both in terms of funding as one of the key element to move things forward. But besides funding, uh, a incubation facilities where you can actually work on your individual product or individual project, like like a large industry working in a mini setup. So in incubation centers, you get to avail all those facilities, the mentorship, access to experts who are well aware of the stages of development, the requirements, and not only the technical but business, IP, regulatory, all dimensions of the of product development and deployment. So we have a footprint of facilitation that is spread all across the country, and we're trying to build the ecosystem in clusters all over the country. So being a central enabling agency, we touch baskets of various elements like innovation funding, uh, academia, reaching out to industry, IP, regulatory, mentorship, access, engaging with large uh, national missions like Startup India, Make in India, National Biopharma Mission, etc. BIREC has actually completed 10 years. It's actually going to complete 10 years on 20th of March. So once we, and it's a good solid 10 years, and we were just running some analysis of the last 10 years. So in the ecosystem, the entire biotech ecosystem, once we compare what we were before 10 years, 2012, and where we are, there is a substantial change. There is a recognition of entrepreneurial uh, potential opportunities. The funding has increased. The number of startups, incubators facilitating them, that have substantially increased. We now have 5,000 startups in the country compared to 50 startups, let's say 2012. The if it's common access facilities, the capacity building has enhanced. We have now 60 BIREC supported incubation centers across the country. Dr. Murthy has one in Naipur, Guwahati, for instance. Like that, there are 60 incubators spread across 18 states. We also have pre-incubation centers. So I think that basket is also increasing in access to the such facilities, of course. There are these startups, entrepreneurs actually churn out their discoveries when intellectually protectable uh, uh, talent that is yielding now the products that have actually reached to the market. So we are looking at good 700 plus products that have actually reached to the market from these 5,000 odd startups, and this along with the ecosystem. This is more towards the initiation of innovation and then deployment. There are larger industries which I have not captured here, which are also increasing, and that product base as well as capacity of both import, export, as well as manufacturing, that is also getting sized up. So we are looking at India's bioeconomy growth, which is increasing in double digit for last several years now. After the 2016, when the Startup India, Make in India national missions came in, so we as India, the India's bioeconomy was 81 billion USD in 2021, and it is poised to grow to about 150 billion by 2025. In terms of capacity enhancements, we support incubation facility, as I mentioned, and these incubation facilities specialize in different subsectors of biotech. For example, for your case, more for life sciences, pharma, biopharma, or related, let's say, medtech. There are dedicated facilities. So these are high-end equipment access, individual working laboratories, or a shared common lab space with access to common access facilities too. If you look across India, there is quite a bit of presence in terms of access to such common access facilities, which have been spread out. At any given time, there are 1,000 plus startups which can seed and incubate in such incubation centers. These incubatees or startups are actually making all of us proud by moving into product development. They have created, seeded their own enterprises, and those enterprises are growing, and many among them have actually deployed their products into the market. So I think that is an ecosystem where we are looking at. And as I was saying, the 5,000 plus startups that we see in the country today, by 2025, we are anticipating that these numbers would grow 2x. So you can imagine the growth and the potential of the ecosystem. And this has actually gained momentum after the action plan for 2000 for Startup India was released by our Honorable Prime Minister in 2016. If you look at the presence of startups all across India, this is sector agnostic. The entire 60,000 plus startups that are registered in India at present, you look at the distribution of such startups, you would notice that there are some places where we have large number of startups which are being deployed, which are registered present, compared to some places where there are lower level of activity, a much smaller extent of activity, and some places you do not have much presence in such entrepreneurial segment. This is when I look at these maps of 2017, 18, and henceforth, these are progressively getting more busier. On the left, if you notice, these 60 incubation centers plus 10 pre-incubation centers, these we are getting populated year on year, adding 10 incubation centers. So this particular capacity building is getting supported, getting enhanced, and it has started churning out in terms of the number of startups also. In the last year, even in the COVID times, we registered 1,200, close to 1,100 plus biotech startups, new biotech startup, which got registered in the last year itself. So these numbers are, as I said, are moving up. I don't know, probably you might have heard about BIREC's flagship scheme, which is called Biotech Ignition Grant. It gives you an excess of up to 50 lakh rupees for a period of 18 months to move your idea to POC, up to TRL3 or more stage. This is, and then you can utilize this funding and go anywhere in any incubation center or a recognized laboratory and work either as individual or as a startup. So far we have supported 700 plus projects under this. These are projects which have at the time of sanction clear demonstrable commercialization potential. Then only this comes and gets considered. Unlike the R&D projects which are more towards exploratory sciences, those are funded by other ministries and departments BIREC supports all funds, all projects that are translatable. The academia is a constant innovation or a constant knowledge generation source, and the industry and academia, if they have to connect and come together, startup ecosystem is one conduit that has emerged out very impressively, and has been able to bring these two together so that the knowledge generation actually leads to knowledge translation and then deployment. And year on year, we are seeing this is getting augmented and scaling up. As I mentioned, even in last year, we saw addition of addition of new startups which got incorporated, and we could see both years of COVID times that was very persistent. The Biotech Ignition Grant BIG, as I mentioned, on the left you could see a graph or India's map reflecting the aspirants for the for this particular grant. It is application proposals come from all over the country. The success rate is just about 10% for them to actually receive this prestigious grant. But I think the important message from this distribution is that there is a inculcation of a culture of entrepreneurship, which is evident in the country. If we look several years back, this map was not so busy, just as we are seeing at present. So this is the power of entrepreneurship, and there has been a fair amount of recognition and inclination around that. One more aspect which we also saw from this, that the distribution of applicants, we are seeing more and more women colleagues are also applying and also getting benefited. So this number, which has increased from 15%, 16% in 2017 to at present, it's has reached to 27% to 73% as a female to male ratios, which is encouraging. As I gave you an example of Biotech Ignition Grant, which is BIG, which provides access to 50 lakh rupees funding. Likewise, as I mentioned, we have created specific customized schemes and programs that are stage driven, ideation to POC, field testing, validation to commercialization. So depending upon which stage your ideation or startup or activity is, there is a funding provision. So we provide up to $1 million per startup access to the fund. And for instance, for clinical trials to de-risk the innovation, which requires much larger funding support. So we support phase 3 clinical trials too, which doesn't have a upper limit of funding. There are, if you look at BIREC's website, you will find there are dedicated schemes for industry support too, besides faculty or individual entrepreneurs or startups. So BIPP and SIBRI, they provide funding support depending upon the TRL level of your development, and the funding support again ranges in SIBRI up to 50 lakh, and for BIPP there is no ceiling, and it supports high fund requirement of particular proposal. So both these schemes have been utilized by industry as well as SMEs and younger startups who are moving into this direction. Excuse me. PACE is a scheme which is more aligned to support faculty entrepreneurs, faculty people who intend to work towards product development, but yet do not want to leave the faculty positions permanently to incorporate their own startups. So academic innovation research, AIR, and contract research scheme, CRS, they are both there to support the faculty entrepreneurship. And the provisions have been utilized by more than 300 plus academic institutions to avail such opportunities. Spursh is supporting social innovation program. That's a dedicated scheme. So there are Spursh fellows as well as Spursh centers who get benefited from this particular aspect. One of the elements is that early stage more funding is given in a grant in aid mode, where if you succeed, you do not have any obligation to return that funding back to BIRAC. Uh, the more advanced stage you are, if your startups are growing, then these are some equity based schemes where some shareholding is shared with the for the particular equity supported grant, just like you would do it to angels and VCs. And then there are follow-on attraction that once you start generating follow-on funds, so angels, VCs also get connected to provide you with the follow-through support. For instance, in these graphs or infographics, you look at seed or leap schemes. About 100 startups that have been supported under these two schemes have been able to generate more than 300 crores as follow-on funding, and 50 plus products have been churned out, and the valuation of these startup companies have increased more than 2500 crores. So this is an encouraging trend that we are seeing, that entrepreneurial journey is rewarding, but it takes patience as well as some difficulties to commit into that. So one aspect is the funding support that is there, but funding alone doesn't take you too far. You actually require a lot of other elements. Compare yourself from to a large industry who would have different verticals for individual activity, for instance, chemistry, biology, regulatory, formulations, clinical trials, IP, etc., admin, all those things, legal, etc. So an individual company or few a group of founders, co-founders would not have that luxury to get associated with all those aspects. So BIREC being a government of India undertaking, we connect with at different levels at G2G, private, public partners, industry, academia, investors, regulators. Uh, so those all connects, all partners become accessible to the ecosystem who gets associated. There is a lot of emphasis on mentoring and high-end skill development for the beneficiaries, and that is done throughout the year through various mechanisms, workshops, hackathons, clinics, and face-to-face, one-on-one, as well as group mentoring. We also bring opportunities for the beneficiaries so that they are aware of the competition, because whenever you intend to get into that that journey, you must be aware of the competition which is evident globally, not only in the local circumstances. So to make India's ecosystem globally competent, there is a conscious effort in that direction too. There are various opportunities like global congregations to reflect India's biotech strength that is put in. Some examples where people who have started their journeys in one small project, and though that product has actually gone to market and deployed. So there are various examples across the biotech sector. You might have been able, some of those probably you might have got exposed to, utilize as well. And that happens both across the best-in-class as well as first-in-class efforts. Being a pharma congregation, I would emphasize that India is largely into best-in-class activities, but I think unless until we move and make more efforts in the first-in-class discoveries and development, we will not become the world leaders or the global champions. So consider once you move into professional careers further, carry that awareness and also make an effort once you have that ability to make and bring a change into the ecosystem. There are some beautiful examples, very encouraging. Deep tech sciences, for example, or ODN related new molecules which are addressing rare diseases by Hanujan Therapeutics, Atenporus, that was licensed out at a very attractive out licensing deal of $125 million. Pendurum is working on liver and corneal tissue generation. The company now has a valuation of more than 100 crores. It all started with a couple of people, just like you students in the IISc. And then today they have moved up, being recognized not only at the national forum, but outside India forum. Ectorius Innovation has been able to create a platform technology that can identify, verify, and uh, study single cell circulating tumor cell at a single cell level from the peripheral blood to gauge and access the reminiscence of metastasis or the tumor. Likewise, in Excel has created WebCare medical devices. So there are plenty of examples. I would encourage you, all of you, to look at this dedicated e-portal called biotech-solutions.com. It currently holds more than 200 BIREX supported commercialized biotech products and technologies. You will get across the biotech sector. So you'll get fair bit of an idea how this uh, entrepreneurship startup ecosystem is evolving. We have also supported manufacturing and high-end characterization facilities. I think everybody would have heard about the vaccine story in the COVID times. So all these facilities, common access facilities, augmentation of vaccine production, characterization, immunogenicity testing, the animal challenge studies, BSL-3 facilities, they have been infused across the ecosystem. The production for ventilators, diagnostic kits, startups, for example, you will moving from diagnostic kit development to establishing their own high-end manufacturing facility recently. So I think there are a variety of examples. If the people who are actually driven in this direction, I would encourage you to follow up and maybe look at some of these, visit the websites and study a little bit more on that. All the vaccines, COVID vaccines that we have seen or got vaccinated, they were one way or the other have been directly supported by BIREC DBT for their translation. Many examples from diagnostic kits, I gave you a couple, ventilators or monitoring devices, remote consultation, PPE kits, there were drug repurposing. So quite a bit of element of activities that had happened that got supported and have been deployed in the market by startups, entrepreneurs, and this includes many from faculty entrepreneurs as well that has translated and successfully deployed. Yeah. Um, yeah, I would, this would be my last slide. I would stop after this. I think this is my request to all the people, both students, faculty who are listening to this webinar. We all understand that academia has a strength in certain areas. It is a center for technical knowledge creation, generation, as well as dissemination. We have access to I'm not saying Naipur Raebareli, but in general, access to high-end central instrumentation facility. You can engage with hospitals, public, private sectors. There's a constant inflow of students from across the country, cream of the talent that comes in. You train every year a batch of students, and it continues, right? It goes on. Um Now, uh, my submission or my request to the faculty and students, kindly encourage visits to, under the curriculum there is provision that industry visits are there. I would encourage you to start considering visit to incubators, because that houses smaller industries. These startups are fall under SMEs, and these are smaller units of of companies. So visit them and see how these things are done at that level. Allow your thesis or dissertation problem statement that can either get converted into a startup after finishing, and there is one particular um, uh, we have created Sitare as one of the schemes that promotes this. Those students who are picking up problems that can end up into a startup, they can get funded, and there is a fellowship of 50,000, 30,000 per year with a grant amount too. Uh, Work with a startup or an incubator that would help you in getting that awareness and orientation towards that. Uh, the problem statement, as I said, one can keep in mind if some percentage of the problems are selected which have entrepreneurial bent of mind. Of course, not the entire academia needs to convert into entrepreneur or startups, but I think some aspect can be considered. Uh, just like we had this today's seminar, would urge every faculty member to engage with at least one or two industries at an individual level to create projects, joint projects with industry. So that will help the associated students, your UG, PG, and PhD students or post PhD. They will align and power up that orientation and would be a great help for their career growth in future. The syllabus, if you can include relevance and significance as well as awareness, what is market competitive analysis, unmet need identification, what is in licensing, out licensing, orientation towards R&D. These are elements when the students go into the industry would find a miss and may be helpful. And of course, the new education policy promotes faculty entrepreneurship in a huge way. So kindly keep that in mind, and there are opportunities. BIREC is one dedicated agency to support innovation for the biotech sector. Likewise, there are other ministries, departments who also support endeavors for R&D. So there are quite a bit of enabling efforts, both at public uh, as well as private sector. So maybe make use of that. I would encourage all of you to do so, and that would actually shape up the next next level of a, a a pool base of the pyramid of innovation. Let's say if we have an innovation pyramid, consider this as a base of the pyramid, and once you succeed the translation and you keep adding, going up the value ladder, and the products, your success becomes the recognition of the ecosystem success. Yeah, I'll stop here, Rakesh. Here. Thank you.
Thank you for the enlightening talk and telling us about the opportunities that are being extended by BIREC for students as well as faculties. Thanks a lot for sharing these things with us. This will certainly help our students and our faculties too in terms of how to apply for different types of fund, because the major thing is maybe people do not know about what are the things that we can apply. That is one of the major hurdle. So thanks for that. And thanks for telling us about the examples which you have shown. I have a short questions on your presentation. Like in your data, you have shown that 53% of funds have been allotted for diagnostics and medical devices. So is it like focus area somewhere of BIREC, or how that comes? Means that's a point that came in my mind.
So the distribution of, it's not a focus area. So this is a outcome of the proposals that we have received and funded. So about 50 to 60% of the total portfolio that we have supported so far, that falls into this segment. This includes diagnostics, medical devices, pharma, biopharma, the healthcare segment, and life sciences.
Okay. And another thing, like as you said during your presentation, the students can be allotted for some project in the incubation centers. If, let's say, for example, if a faculty or a student is interested to do his own work on his own idea in an incubator of BIREC. So how can we apply for that? Can we apply through online, or how is that?
So all incubation centers are open access, common facilities, common for capacity development as well as human resource development. So the students, entrepreneurs, faculty, any stakeholder, they can reach out to these incubation centers. We have 60 incubation centers and 10 pre-incubation centers across the country. These get more are getting added. So the details are there on BIREC's website, as well as any of these incubation center also have their individual website, and there is contact details of the person. You can walk in, okay, and anybody can walk in, connect to them. There is dedicated incubation manager, CEOs. They would be very happy to answer your queries. Even if you walk down with a very simple looking query, concern, or curiosity, you just go speak to them, understand the system. They would be happy to share and give you an orientation and answer the questions. And probably, and so one more thing I would like to say is that incubation centers are the focal point. Right. For example, if BIREC is coming out with a new opportunity, we would of course spread out and becomes like we share that information all over India. But mostly this gets diffused out through the incubation centers. So these get channelized. So incubation centers are the focal point for not only single opportunity but all possible opportunities. They keep a tab also. They're the most current, and anybody reaching out, they will be happy to share the details.
Okay. So that's a very good and valued suggestion that we should visit those incubation centers to know what is going on there and how they can help us in terms of students project or in terms of some idea developing into a product. Thank you for extending all these talks to us. Any questions from the audience to Dr. Diwan? He would be happy to answer. From students or from faculties at NIPA Library. I think Dr. Nimesh is going to ask something.
Yeah. Sure. Yeah.
Thank you, Rakesh sir. So first of all, Manish sir, hope you're doing well. So long time no see. So I have a short question in connection with what Rakesh was asking. So this was regarding, so let's say from the industry point of view, because to be frank, I am mostly been in the industry. So if a student wants to get into an industry, so does BIRAC support anything like that? I mean, we know I get a lot of people from BSIL, BCIL trainees and all those things. So is that a part of BIRAC or is something, if you can just let us know? Yeah.
Yeah, sure. So BCIL is one such provision that has been created through Department of Biotechnology, which is our parent department, that provides fellowship for the trainee who is inducted in the industry. So that's a one specific arrangement. BIRAC directly doesn't provide fellowship or directly ask any industry to pick up somebody. Conventionally, that has been the case that students try to reach out to industry for internship or fellowship, no, for the trainings. The industry footprint in India is not very large. Not all industries are doing R&D, and R&D, as we all understand, is very cost intensive. So they would not be happy to share their resources, reagents, access to the validated QA qualified instruments, to students who are not trained as yet. So they can be, few of them allow you, but be observers. Instead, I am suggesting that also keep an option to access to startups. So each of these incubators would have a varying number, maybe 25 to 60, 70 startups seeded in the incubation center. So you imagine those many number of new opportunities, even for one single incubator, right? So they will be happy to have young students doing little bit small defined activities as any industry would have wanted. Nimesh, we do not have direct infusement for fellowships for the students, but through the various funding schemes, the provisions, if availed, they become more tuned, not only to create their own industry, because we create job creators rather than job seekers. But in tandem, they can still be industry ready if they tread that path. Thanks.
Thank you, sir. Thanks, sir.
Any other question from any of the audience? So I think for now there are no questions, and if there are any questions, I will get back to you with their questions. Maybe I can mediate those questions for you, sir.
Sure, no issues. I think it's been a long day. I see students would have been sitting. I looked at the agenda. I felt that it was an overdose of highly concentrated, high value information. But I think it's an excellent effort. I'm very glad that many of my previous colleagues have been able to spare time and join. And I'm also happy to engage. Thank you so much. Thank you, everyone.
Thank you, sir. Thank you so much.
Bye-bye. Bye then.
Bye-bye. So I think we have come to end of the session for the day. And before closing, I need to conclude. So thanks to all the participants who has participated, and I would do it formally with concluding remarks. So respected director, sir, distinguished guests, and invited speakers from the industries and public sector organization, participants and colleagues from NIPER, Raebareli and other institutes, and dear students. It gives me immense pleasure to propose the vote of thanks for the organizing today's symposium event, which was like the industry perspective on translational challenges in drug discovery and development. We listened to the valued talks delivered by eminent industry experts and got an opportunity to learn about the industry quest to get the right target with the right efficacy, safety, and evaluating them in right patients with right commercial potential. And the challenges faced by pharma companies in this quest were shared by them. What are the challenges associated with it? During this symposium, we had an opportunity to discuss all the challenges in translational research to learn newer, many innovative approaches, how we can develop to overcome those translational challenges. I'm sure the audiences must have been benefited from the diverse and rich experience of the experts. And this symposium has provided us new insights to face those translational challenges of discovery and development. So I highly appreciate and thank the invited speakers who have agreed to come and deliver their expert talks to share the wisdom and knowledge they have. Thanks and congratulations to all of you for the successful symposium event today. Thank you. Thank you all.